5 बजे उठा दिया है।
मैं समझ रही हूँ तुम्हारा क्या टाइम हो रहा है। तुम्हारा बज रहा होगा 10:30, 11:30, 12:30, 1।
नहीं नहीं नहीं, मेरे यहाँ सुबह हो रही है, 6 बज रहा है इस समय।
कहाँ पर हो आजकल तुम?
मैं जर्मनी में हूँ, तो 4.5 घंटे
जर्मनी में हो?
पीछे चल रहे हैं आपसे, आप लोग आगे।
अच्छा, मुझे जहां तक तुम यूएस में हो उस हिसाब से मैंने बताया। तो सुबह से तुम उठ करके बैठ गए हो?
मैं 5:15 से बैठा हूँ।
अच्छा।
मदद हो गई राइट टाइम होने की ना अब।
कब से नहीं आए तुम इंडिया?
मैं हो गया मुझे। लास्ट ईयर आया था, मतलब पहली बार बहुत गैप किया था, 2-3 साल का गैप किया था, उसके बाद लास्ट ईयर आया था, अभी फिर आ रहा हूँ, अगस्त में आऊंगा एमिटी।
देखो एक अब जमाना इस बार तो मिलना। देखो उधर से श्वेता भी झांक रही है। क्या हाल है श्वेता? ये तो यही है, उससे मुलाकात नहीं होती। हाँ। हाउस मैम, आप कैसी हैं? हाँ श्वेता, कैसी हो? मैं बढ़िया हूँ मैम, आप कैसे? बढ़िया। चलो अच्छा लगा, आज तुम लोग को सुनेंगे हम। कि आज हमारे स्टूडेंट्स, हमारे बच्चे बोल रहे हैं, अच्छा लगे सुनने में। थैंक यू मैम। ठीक है, चलो अब शुरुआत होती है। हेलो श्वेता। हेलो कृष्णपाल। हेलो मैम।
हेलो मैम।
गुड बस मॉर्निंग मैम। कृष्णपाल, आई एम सो सॉरी, तुमको सुबह सुबह जगा दिया। बस स्टार्ट करा रहे
नो
हैं।
प्रॉब्लम।
क्या इंटरनेट, तुम तो यू बोथ ऑफ यू आर अवेयर अबाउट द एमिटी सिस्टम। बस
या
एकदम
या
2 मिनट का टाइम, बस 2 मिनट में
ओके।
स्टार्ट। श्योर मैम, डेफिनेटली। थैंक यू। अब तक ज्योति मैम से आप बात करो, बस 2 मिनट में ट्रैक करा रहे हैं।
ओके।
श्वेता, तुम तो आईआईटीआर में ही हो ना?
नहीं मैम, मैंने कॉर्पोरेट ज्वाइन कर लिया इन सिलिको माइंस हैदराबाद। अभी
आवाज
मैं
चेक कर रही हूँ, कहीं
एक
प्रॉब्लम
साल
है?
हो गया मैम यहाँ पे।
फिर से बोलो।
और मैंने कॉर्पोरेट ज्वाइन कर लिया है।
अच्छा।
पीएचडी प्रीवियस ईयर कंप्लीट हो गई थी, तो उसके बाद इन सिलिको माइंस अभी सीनियर साइंटिस्ट पोजीशन पे हूँ यहाँ पे।
अच्छा।
और yeah,
श्वेता, आपने शायद पारसारथी के साथ किया है ना पीएचडी?
yes मैम।
इस्माइल, जर्मनी में कहाँ पर हो?
हम जर्मनी में रोस्टॉक।
रोस्टॉक। रोस्टॉक यूनिवर्सिटी में?
हाँ, रोस्टॉक यूनिवर्सिटी में।
अच्छा।
ये सिटी बस यूनिवर्सिटी है।
अच्छा। जानवी, आई थिंक यू मस्ट हैव टू स्टार्ट द सेशन। हेलो।
Good morning, everyone. We welcome you all to the second day of our workshop, where we continue our journey of insightful discussions and sharing knowledge. Today's session are lined up with distinguished speakers who are expert in their respective fields, bringing valuable insights into the world of bioinformatics and computational biology. So, we would like to felicitate our first speaker, Dr. Krishna Pal Singh, sir, with an e-booking. Now, I would like to felicitate our second speaker, Dr. Shweta Singh Chauhan, ma'am, with an e-bouquet. Now, we would now like to felicitate our third speaker, Dr. Jitendra Narayan, sir, with an e-bouquet. Before that, we are honored to have Dr. Shalini ma'am and Dr. Meenakshi Srivastava ma'am as the esteemed judges for this event. Their expertise and insights with greatly enhance the discussions and evaluations throughout the sessions. To begin, with our honored to introduce the esteemed speakers of the sessions. Our first speaker is Dr. Krishnapal Singh Sir, a renowned scientist in the Department of System Biology and Bioinformatics at the University of Rostock, Germany. With over 11 years of expertise in bioinformatics, system biology, molecular dynamic stimulations, QSAR pharma co-food modeling, and RNA sequence analysis, Sir has significantly contributed to the understanding disease mechanism through computational approaches. His research in the MEL-AUTISM project focused on the crosstalk between melanoma and autoimmune disease using system biology-based disease map approaches. His work integrates the computational strategies with experimental data to identify novel therapeutic targets, helping shape the future of precision medicine. And our second speaker is Dr. Shweta Singh Chauhan ma'am, a senior scientist at In Silico Minds in India. She holds a PhD from CSIR IITR Lucknow and specializes in modeling and simulation for AIP synthesis, molecular modeling, dynamic simulation, and structure-based drug discovery. Her expertise spans computational biology, toxicity assessment, and biomarker identification. She has contributed extensively to advancing in silico methodologies for accelerating drug discovery and development. Her research focuses on AI-driven predictive models for drug interactions and toxicity, playing a crucial role in pharmaceutical industries' move toward more efficient and precise drug development strategies. Moving forward, our third speaker is Dr. Jitendra Sir, an esteemed expert in bioinformatics and data-driven biological research. With extensive experience in computational methodologies and their applications in biomedical sciences, Dr. Jitendra has made significant contributions to the field. His research has been instrumental in advancing computational tools for genomic analysis, facilitating a deeper understanding of genetic variations and their implications in disease progression. He has worked extensively in integrating multi-omics data for precision medicine, enhancing the accuracy of disease diagnostics and therapeutic target identification. His expertise spans systems biology, artificial intelligence in healthcare, and big data analytics, leading to numerous high-impact publications and collaborations with global research institutions. His groundbreaking contributions in computational genomics have opened new avenues in personalized medicine, offering innovative approach to understanding complex diseases. Thank you so much, sir, for enlightening us on such an interesting topic. With that, I would now like to hand over the session to Dr. Nikita Basant ma'am and Dr. Jyoti Prakash ma'am as the chairperson for this session, who will be leading the discussions and moderating the talks. Assisting in documentation and key insights, we have Dr. Osaf Ahmed sir and Dr. Rachna Singh ma'am as the rapporteurs. With that, I would now like to hand over the session to our respected chairperson, Dr. Nikita Basant ma'am and Dr. Jyoti Prakash ma'am.
Thank you, Rida. So moving on, I would now like to welcome Dr. Krishnapal Singh, who will be talking about the role of systems biology in target identification using disease maps. So without much delay, I would like to now invite Dr. Singh for his talk.
Yeah, thank you, Nikita Basant ma'am and Jyoti Prakash ma'am. So first I will, please give me permission so that I can share my screen.
Sir, you have the permission.
Okay, now it's okay. Okay, my screen is visible?
Yes, it's visible.
Okay. First of all, I sincerely appreciate the invitation of the talk for the bioinformatics event, and it is a wonderful opportunity for me to share my work with the METN. So, begin on my being on an METN, I have completed my master's and my PhD from MIT, and I also serve as an assistant professor in MIT University as in a research cell. So I have gained a lot of experience from the MIT. So I want to share all my experience, what I have learned from here in Rostock University and disease map and system biology I can share with you. So whatever the topic you see on the screen is role of system biology in target identification using disease map. So when you are talking about the system biology and bioinformatics, so these are very well connected because our biological system is like is a complex system. So to understand the complexity of the complex system, you have to make a proper system so that you can understand the communication between the any nodes one to another and another to another. So you have to make a network kind of thing So when you are making a network, when you connect all these network, it make a system so that they communicate one system to another system. And for to identify the target in between these systems, the communication between the systems, so you have to make a proper network or disease map. So I will show you in the coming slide how you can make your system for the disease map analysis. So first of all, in University of Rostock, what we are doing in our department is the data management. In the data management department, information retrieval from workflow and development and data science like machine learning, deep learning, and image analysis also is a separate department for in our lab. And network modeling, what I will show you in disease map from large scale network to the core regulatory and the single molecule. And molecular analysis is docking, simulation, pharmacophore modeling. And all these things, when you merge in one platform, then it comes to the applications like drug development, clinical diagnosis, and personal therapy, and understanding the mechanism. Here you can see in this picture where there is a large scale disease map. Means when you are dealing with any kind of disease, you have to understand the mechanism. You have to start from some phenotype, means collecting the phenotype and the lead molecules. So when you have in your mind, like, okay, this disease is started with one mutation, like lung cancer is started from BRAF mutation. So you have some lead molecules. This is the base layer single molecule. And when you have a phenotypes, which types of cells are involved in the disease condition, then you have a large network also, means lot of cells are communicating in the disease condition. And in between that, there is a core regulatory network. So in our department, we are starting from the large network and coming to the single molecule. And when we got the single molecule, we can perform different types of analysis like docking, simulation, and other mechanistic thing. Why do we need in silico network models? Here you can, in this picture, you can see, as I told you, the biological system is a complex system. So it starts from the higher level to the lower level. So what is the higher level? Higher level is the organ and tissues. You can see in this picture, when you have a disease condition, you get reflection on this layer, means organs. You have your organ is not properly functional, or some problem in this layer. So it means the problem in the top layer arise from the lower level in the molecular network layer. So if you are talking about the system biology, so you have to understand complete network from the lower level to the top level. And these three layers are communicating with one another. Means if you start from the organ, then it comes to the cellular level, how the cells are interacting, and it's come to the molecular level. So in our disease map approach, we are designing three different layers, starting from the organ layer, then the cellular layer, and then the molecular layer. Why we design three different layers for the disease map? Because the clinician are interested in the organ level features only. They are not interested in the molecular level. So they only look for okay, this organ or what types of function or dysfunctioning or what are your problem in the organ level. So for the clinician, the top layer is very important so that we can express or we can just communicate with the clinician. And the second layer is the cellular layer. Means when you are in the lab in vitro, they are only focusing on the cell. Means in this cell, which gene is overexpressed, underexpressed. So for the cell, for the lab purpose, it is very important the cellular level, and the molecular level is for the bioinformatician. So that you have one lead molecule, you can inhibit that one, and you can say, okay, these red dots are the default or the mutated proteins you can inhibit, and you can see the impact on the cellular level, and then it will reflect on the organ level. So when we start to design any disease map, we can design three different layers. I can show you how it will come into the reality. So first come to what is simplified pathway model. Simplified pathway model is when you modulate S and then upregulate S and it modulate pathway X. Means simple, linear relation between when you are upregulating S, then it will go impact to the X pathway. But as you know, the biological system is not simple. It's very complicated. So this is the one example of the biological system where cells and cell are communicating with one another, whether it is healthy patient or it's a disease one. So when this communication get disrupted due to any mutation or any problem, then the disease condition arise. Here you can see the lot of cells are interacting with the different types of chemokines, cytokines, and these types of communications. But it is not, it is you can say a cellular level. And when you go to the molecular means layer, then you get a pathway. Means every cell has its individual pathway. And in this pathway, you found a mutation or any problem. So when you found any problem in this cellular pathway, it will reflect on the cell level, and then the communication will disrupt it. So it's when you see the complete picture of this scenario, you found that it's a bit complicated because you have not focusing on the cell level only. You have to focus on the pathway, and in the pathway you have to focus on the particular protein, how they are communicating inside the cell. So now picture change. Now if you modulate S, what will happen? Then there are different pathways are modulated, as I shown you in the previous picture, and it is not a linear relation. Sometimes it's inhibiting the pathway, sometimes it's activating the pathway. So there is fluctuation in the pathway, and you cannot predict the function of any pathway in the realistic, because it's a dynamic process. Sometimes it's up, sometimes it's down. So you have to capture all these time frames also, because time frame also plays an important role in the disease map. So inflation, we started this project with inflation because it's very complicated and simple. You can both thing you can say. So we design on the blackboard some cells, putting some cells how they are communicating by reading some books and some papers, and we put on the blackboard these cells. And then with the clinician, when we went to the clinician, they make some input also on this picture, and then we add some more cells and clear-cut pictures so how their cells are communicating. This is the top layer, the top layer we are framing. And after that, we design a graphical interface for this inflation, how their cells are communicating with one another. And each cell has its own pathway inside. When you click on this, I will show you the real picture of this, this project also. So when you are click on this, any cell, then it will go inside the pathway level also. So this is the final version where we start with the, wait a moment, where we start with the initiation and the transition, then the homeostasis condition. So it is complete process from the inflation to overcome the inflation. So all the cells involved, all the processes we capture within the cells. Okay, as I told you, when you click on any of the process, then it will zoom into this, and you can see here, each cell contains different communications of the nodes. So this is system biology, what I am talking about here. Means when we design this cell, and then inside the cell, we make a complete pathway of communication between the nodes. And there are different phenotypes. So if you change any of the concentration of any of the nodes inside these cells, it will reflect on the phenotypic layer. Means at the top layer, you can see the impact of these changes on the nodes. And what is the next is workflow of the disease map construction, how we design this disease map. So coming to we adopt two approaches. One is the top-down approach, means starting with some phenotypes, means we have to collect some processes, some cells, which is involved in the disease condition. So we collect all the information related to the cells, which are the faulty or which communication break, all kind of things we collected in the top layer. And when we have this information at the top layer, we just zoom into the cells and make the pathways inside each and every cells and how they are communicating with another. And with the help of Minerva platform, we uploaded all the disease maps into the Minerva platform so that anyone can visualize, anyone can analyze his own data. I will show you in the coming slide how it will work and everything, but it is a basic phenomena. And when we are talking about the top-down approach, means we are starting from the top and going to the bottom. But what about the bottom-up approach? So we adopt in the second phase bottom-up approach. In this bottom-up approach, we collected some key molecules like lung cancer. We have a BRAF, MAPK. We collected all the mutated proteins and started designing and growing these nodes, how they are communicating with the other proteins. Means with the string, we make a network, but the problem in the string is they are not directed. So we have to make the directional of all the nodes, because when we upload any kind of data like omics data, we have to check the impact on the phenotypic layer, and it will come when we have all the nodes directed. So we have to direct all the nodes by reading the research paper from the different databases like Reactome, KEGG, different databases, and then we make a communication between these nodes also. But it is not complete story. We have to add one more layer, that is MIM, molecular interaction map. We make a script, we made a script and extract all the data, which is from the different like miRNA, TAR, miRNA, drug base, and Reactome. We fetch all the data which contain miRNA, lncRNA, transcription factor, drug, and metabolites, and merge into the top-down and bottom-up approach and come to the inflammation disease map. So after merging all the data into our disease map, we have 6,000 elements, nodes or molecule, and 20,000 interaction. In this picture, you can see it's look like this. It's a bit complicated. This is the real system biology, where you have to identify the core regulatory network in this network. So this is the task. So here I can show you the clear picture, what from where we start and what is the end. This is the first layer. This is the phenotypic layer. 42 cells we are collected, which is contributing to the inflammations. And the second layer is the process layer. When you dig into the cell, you get a pathway. So we design the different pathways in this phenotypic, in this process layer, and 40 submaps, and 2,226 elements, and 2,367 interactions in this layer. And the bottom layer is the molecular interaction map, where we included the miRNA, lncRNA, transcription factor, so molecular interaction map. So this is the architecture of disease map. You can start from anywhere, means from the molecular interaction map to the phenotypic layer, or you can start from phenotypic layer to the molecular interaction layer. So I will show you from starting to disease map how you come up with the lead molecule. So identify the molecular data, means you have the cellular information. You start from that point or the lead molecule, and then create a disease map, means you are, as I showed you, you have to design the 3 layers, and then curated the submaps, which is very important for the submap, means every cell has a pathway. This pathway is a submap. And then upload all these map to the Minerva platform so that everyone can visualize and can do the analysis for their own work. And you can map any omics data, whether it is array data, RNA-seq data, whatever data you want to map, you can map on the disease map. And then after mapping the data, because in our disease map, there are 2 types of plugin. One, without data, it will analyze your regulator or the lead molecule, which is based on the topological parameter, because nodes are connected, degree and centrality and betweenness, all these parameters are inside the network. So based on that, without data, if you are analyzing, it will give you the lead molecule based on the topological parameter. If you upload your own data on the disease map, then it will analyze your data and then give the lead molecules. So we have both options, means without data and with the data. And then you identify the proteins, which is very important, and calculate the influence score. Means each proteins, when you are uploading any data to our disease map, it will generate the influence score for each node. Node means protein. And based on that influence score, you check, okay, I have uploaded the melanoma data, and based on this melanoma data, these proteins has a higher influence score because they are communicating in the background. And when the higher influence score, you can extract that proteins because they are highly impactable by this melanoma case study. So then you identify the regulatory motives. But if you have 10 regulatory motives, so you have to perform molecular docking. You cannot perform 10 molecular docking and simulation. It's a big task. So for that, you have to prioritize the target. How you prioritize the target? Then it's a Boolean modeling or mathematical modeling. So that's why I showed you here is the prioritization of the target. Based on the Boolean or mathematical modeling, you can prioritize your drug. Means perturbation experiment. I will show you how it will work, the perturbation experiment also. And then you identify with the lead molecule, one molecule, then you perform molecular docking and check the stability, and then the lead drug for the disease map or for the disease. So in our disease map, there are two types of input we are taking. As I told you, first one is the publication and literature. You can just make based on these data, we are designing our disease map. But you can, the newly generated data means RNA-seq, omics, whatever studies in the public domain, you can also map these study to our disease map. And based on these two input, they are making they are making a hypothesis. And based on this hypothesis, we are generating a mathematical model, prioritization of the drugs or the lead molecule. That is the mathematical model. And again, is checking the hypothesis and generating a new hypothesis. And this hypothesis will be tested in the lab and it's worked perfectly. Then we can say, okay, our disease map is very similar to the what disease is saying, or disease is communicating with us. So for this, first I will show you how it will, in real scenario, it will work. I will show you in the real scenario. This is on a disease map. Yeah. I hope my screen is visible now also.
Yes, it
it's
is.
visible.
Okay. So this is one example we have just recently completed is a nose OE project is for the COVID patient. We have just uploaded the COVID patient data. You can see here is the different, some maps are there. Yeah. So sustentacular cell, horizontal basal cell, all different cell. When you click on this, it will show you the inside of the cells. Yeah. So when you go to this design and you click any of the map and it will show you the communication, how their cells are communicating inside this layer. This is the primary version. Now we have to just modify more, but at least you can see everything. When you click any cell, then you can go inside and check this is the node and it's complete information here. Binding affinity, which protein is bind, from which literature it's coming, and from where we have taken this information. Yeah, and everything of this node is here. And you can also do one thing in this. It's very and one thing. This is the explore. If you have no data for the analysis, you can just put any of the data here. I can show you. And here is the data. You can map any RNA-seq data or omics data or array data, and it will generate a result for you. So I will show you one example here. Enrichment. Here you can see, wait a moment. You can add any node into this, like CRB1. Here you can see, you can click and it goes to the original paper from where it is extracted. So everything is documented here and no need to worry about from where it is coming and how we make the connections. And also I will show you one thing very quickly. Yeah, if you add any node like CREB1, if you are interested in this molecule, wait a moment, downstream. Yeah. If you added this molecule, means for time being, if you are interested in this molecule and you know the information that this is the faulty gene and you have to overcome this gene for the disease condition. So you can add this into the perturbation experiment and you can downregulate this gene. Okay. What is the impact when we downregulate this gene and you have this in the phenotypic layer, you can see the impact of this gene in the real time. And you have, if you upregulated this gene, and then what will be the impact on the phenotypic layer? So every information in this disease map you can see with this simple change in the one molecule. And you can add number of molecules here, 10, 20, 50, and you can modulate in real time and check the impact of this particular one modification in the gene and what will the impact on the three layer, both means top layer, middle layer, and the bottom layer. Now coming to my slide. I hope it's clear the things what I am telling. And with this, the network curation means like this type of network. Inside this, you can upload your scientific literature and molecules. Based on this, we have created this one, and you uploaded like I show you in this real scenario. You can upload any kind of experiment to our disease map, and then it will make a perturbation experiment. Means you can change any molecule, and it will show you the upstream impact, downstream impact, and the signal progression also. So what types of research question answered by the disease map? First one is the disease molecule association. As I showed you, means if you change any molecule, then it will show the impact on the disease. And predicting the impact on the phenotypic layer, it will change the phenotypic layer also. Finding the diagnostic and prognostic marker also, and the therapeutic checkpoint also. And from which data you can map clinical phenotype, cell type, or a specific molecule or omics data also. And this is the standard SBN, SBG and SPEN format of the disease map. And analysis you can do. The disease-specific co-regulatory network you can find, pathways, enrichment, and the patient classification, and this and drug target prioritization also you can perform with the disease map. So for this, as I told you that we have two type of plugins. One is the data dependent, second is data independent. Data dependent, if you map, only we need the gene ID and the log to fold change. Simple, very simple. Means you have to upload the gene and the log fold chain, and then it will create inside the map, it will create an influence score for particular gene 1. We have influence score this one. And the metabolite this, and they have an influence score this. So it will create an influence score. Based on this influence score, you can prioritize the nodes of your network. I hope everything is clear. It's a bit complicated, but it's working like very simple. When you use this portal, it's very simple. You have to just upload your data and analyze the things. Like I showed you this in the real scenario, like Explore and Omics plugins we have, and you can upload, and whatever the nodes map into the disease map, it will show like 2 to 9 elements was mapped directly to the map. So all this information, and it will generate an input for everything. For one case study, I map one RNA-seq data, which is this idea of this data. This is the melanoma metastasis. In this case, there are two control, and rest is the melanoma metastasis cases. So I extracted with the RNA-seq data, I extracted the differentially expressed gene based on this data. And now I have to check whether the melanoma is communicating with the autoimmune disease or not. There is any crosstalk between melanoma and autoimmune disease. So I have to find out this answer of this question. So that's why I just created a melanoma disease map connecting to the autoimmune disease like rheumatoid arthritis and ulcerative colitis and Crohn's disease. So now I have to upload the data and check whether my disease map is working fine or not. So I just put data of this case study into the Omics plugins and just uploaded, and filter is 0.5, p-value, very simple. Whatever you want, you can do in this portal. And then we found that the rheumatoid arthritis phenotypic layer is 1.5 fold up, and ulcerative 1.3, and Crohn is 1.4. It means there is a communication between melanoma and autoimmune diseases when the checkpoint inhibitor therapy is going on. It means, and we found a lot of paper also. So that's why we are curious. We have to check whether these papers and our disease map are working in the same scenario or not. So that's why we checked this one, and we found this communication. And what are the next question is coming to the picture? Which genes or which proteins are communicating from melanoma to the autoimmune disease? So this is all the mathematical thing. I am not going into the detail of these things, but this is the common genes which are communicating from melanoma to the autoimmune diseases. And as I told you, I showed you the influence score. So we have generated influence score of all these proteins. Here you can see, and this is the melanoma log fold change, log 2 fold change. So when we map this log fold to our disease map, then this influence score are generated, and these are the common genes which is communicating from melanoma to these autoimmune diseases. So after that, with our disease map, we check the upstream process, means we have to check the master regulator, whether any protein or genes are regulating all these common genes or not. So we just go to the upstream process, not to the downstream, because we are interested in the master regulator. And we found there 3 master regulator of these genes. Not 100%, means 50% they are capturing this common genes. One is SP1, and the second one is PP3CA, and this is Malta 1. This is the LNC RNA, because in our disease map, the transcription factor, LNC RNA, everything is present. So this is the LNC RNA. So we drop this LNC RNA for our analysis. And then now we have 2 targets we have to inhibit. Now the bioinformatics come into the picture, molecular docking and simulation, any kind of thing. Huh? So for inhibiting these 2 targets, we have to check if we inhibit these 2 targets, whether they are contributing to melanoma or not. Or if we inhibit this target and they are not contributing to melanoma, then it will a problem in the network. It will arise it will just generate some other disease also. So we just we went to the TCGA data and check whether the SP1 and PP3CA is contributing to melanoma or not. And we check expression. So both the genes are contributing to the melanoma, means in the melanoma metastasis, they are upregulated. So we can use both the target to inhibit, because if we inhibit these 2 targets, they are also contributing to melanoma, they are inhibited, and also the common genes. And our objective is to cut the crosstalk between the melanoma and autoimmune disease. So we also check the survival curve also for these 2 proteins. Huh? Whether what is the survival probability when they are upregulated. So when they are highly expressed SP1, the survival probability of the patient is low. And same is the PP3CA also. So we are very much confirmed that, okay, they are overexpressed in melanoma, and when they are high expression, their survival probability of the patient is also low. So these 2 are the perfect target, and they are targeting our common genes also. So based on this, now we come to the bioinformatics structural biology part, and just went to the FDA approved drug, because we are not proper clinical lab or experimental lab so that we can perform the experiment. So we have to play safe side. So we just select the FDA approved drug and start the virtual screening and molecular docking and MD simulation for against 1,615 drugs, which is FDA approved. Now maybe the data is always changing. So, and we found like one drug is metanoic acid, has a docking energy of -142 for this. and SP1 and PP3-ACA, we identify another drug. So the line is that we started from the large network. Now we come to the single molecule. So I hope you understand means from where we started and how we are reducing the target and coming to the single molecule. And in the single molecule, how we prioritize the one key molecule. And then when we prioritize the key molecule, then we perform the docking simulation, any kind of analysis. So this is our team in SBI, and these are the collaborator which I am working in this project. And I hope this everything related to disease map. If anyone has
Thank you,
any
Dr. Singh.
Yeah.
That was a very interesting talk. And I think that we as bioinformaticians, we are very often working with the bottommost layer of the molecules and drug protein targets, but we very often want to understand how it translates into the bigger picture, if you're making an impact in the bigger picture or not. So it was a very well laid out presentation. Now I have two questions. The portal that you're using for developing these maps, is it something that your lab has developed, or is it a commercial software that you're using for this?
No, we are using that cell designer we are using. And the only we are using one portal, which is I think a TU Minuk platform is Minerma to just display our disease map. Only all the rest thing, the plugins and everything, disease maps are created in-house. Only for the visualization, we are using the Minerma platform, which is from the TU Munich, and we have a collaboration with them.
Okay. Okay.
Yeah.
And I believe that you mentioned that you're also using some mathematical models in the entire coursework.
Yeah.
So that I believe that means machine learning, AI and machine learning. Were you talking about that?
No. Machine learning, we have to implement this machine learning to make it automatic, because we have lot of, because in the Europe, there is lot of problem in collecting the clinical data, because we are always suffer these kind of problem, collecting the patient data. There are very much restrictions. So we are generating a synthetic data. Our department is a machine learning department, is a separate department in our lab. They are generating the synthetic data, and then based on the synthetic data, we have to make its mathematical model. But for this disease map now, I am talking about the mathematical model like prioritization of the drugs. Only we are designing the mathematical model like Boolean model. Means one is activating the drug and what will the impact on the whole network. And when we off the target, then what will be the impact? So that is the mathematical model. This, in the simple word, you can say a Boolean model. Yeah. At the background, there is a
On and
Boolean
off
model.
switch.
Yeah.
Okay. All right. Okay. So thank you, Dr. Singh, for such an intriguing talk and such an interesting research that you're doing at your end. So moving on, I would now like to invite
Nikita ma'am, I also have one question, very general question. Krishpalji, very general question I have. What are the major challenges when you are using system biology for target identification? So what do you see the major challenge behind this?
Major challenge is one when we are presenting this our disease map, because lot of companies is using our disease map, like Heal is the pharmaceutical company, Europe biggest pharmaceutical company. They are using and they are patenting one drug also based on this our disease map. But the problem we faced with the pharmaceutical companies, they always ask, like, when we mapping the data, any kind of data in our disease map is bulk data, means RNA-seq data. And the problem is that if we say this A gene is upregulated, so they ask for from which cell, then we have no answer, because it's a bulk data and our disease map only handle the bulk data. Now, when we realize this kind of problem, because the clinician focus on the particular cell, they want the name of that cell. If A is upregulating, they ask from M1 or M2 or T cell or B cell, from where this A is coming. But from our disease map, we cannot answer. We can only answer that it is upregulated in the disease condition. So they are interested in particular cells. So we are facing this kind of problem. That's why we are just modifying our disease map so that in the next version, we clearly say if A is upregulated, it is coming from macrophages. It's not from T cell. So if you have to inhibit this A, so you have to work on the macrophage, not all around the cells. These are the some problems, and it's always upgrading these disease map is yeah, it's a continuous process.
Thank you so much for nice presentation and for solving my query. Although query is very general, but I was able to understand what is the system biology, how we can use, or what is the uses of the system biology for to target or the identification of the disease. So thank you so much,
Thank
Pushpalji. And dear audience, if you have any question or any query, then you can ask. Our expert is here, and you can get knowledge. Dear audience.
Yes, I have a question. KP, how are you?
Yeah, I am fine, ma'am.
So, KP, whatever study you have discussed till now is from the data that you have retrieved from non-Indian population, right?
Yeah. I have now because whatever I did the analysis with the melanoma and the COVID, so it is independent of this. The only I focus on the patient and not the reason wise. Yeah.
Region wise is not, it is not separated region wise, just you are focusing on the disease
Disease,
and the
yes.
patients, the
Yeah.
data from the patient wherever it is deposited.
Yeah.
Okay. So your main work was on COVID only. So right now, what are the focus of your lab? Because now COVID is over, still work is going on, just for information only.
So basically, our lab is focus is on the disease map, no? So it's independent like COVID, melanoma, or some other disease, rheumatoid arthritis. So because we are working with different companies here, so they are providing the disease target, means they are saying, okay, we are have this kind of disease. Like as an example, Heal. Heal is a pharmaceutical company. They are making a drug for the athletes in German athletes, no? So they are focusing on the herbal product only, not the chemicals. So
Okay.
they have a means athlete has a different kinds of problems, na? So whatever the rare disease also is included in this. So whatever the disease, we have started to design the map for the disease and come up with the target and serve for any kind. So we are like a service provider, not
Right,
not a
right.
single disease.
So, uh, so that would you find that is it difficult to work with the herbal products in comparison to chemical products?
Yeah, because and I cannot say which herbal product or chemical product, but it's difficult to work with the companies because the companies have very strict guidelines and they will not give you any chance to go right and left. Whatever they want, they want. Na? And they have a lot of question like in herbal product. From which plant? Because I am not a botany student, so whenever I am went for the meeting, so I always face a problem because with the name, lengthy name and everything, and they can ask any question na, at the real
Right.
time. So I face this kind of problem. I am very much friendly working with this cancer and all these things because now we have one new project is melanoma heterogeneity. It's a new topic. So we are working and DFG is funding also for us. So I hope we come up with these things. But herbal product and botany is weak point for me. Only I
Okay, no
understand chemicals
problem.
and molecules.
No
Yeah.
problem. No
Yeah.
problem. Okay, thank you so much. All the best.
Thank you, man. And thank you to all for AIB faculty member because I spend a lot of time there. And in August, I will be in India and I will visit campus also.
You never visited us. I saw your
One time I
post
visited
continuously.
when I have to do
Hope to
PhD.
see you. Hope to see you next time.
Yeah. Thank you, ma'am. Okay.
Okay, thank you so much.
I have a small query. KP, many congratulations. It's a nice presentation. Although I'm not the expert of this field, but I have just one query.
Yes.
When you prepare these disease maps, you will use the data which is available at that time. You will input the molecules which are discovered at that time, isn't it?
Yeah.
And then you interpret from that data.
Mhm.
Maybe at that time, some other molecules are also involved, but those molecules which are available at that time, which are published, you are preparing disease map from that data. And when you take the interpretation, don't you think that will be somewhat biased? In a way, whatever data is available, the result will also be like that. It is not the actual scenario. It is the scenario which is available till date.
Yeah, till date. So that's why we are, whatever we are collecting in our disease map, whatever the connection we are making, we put the PubMed ID also, so that it will be become clear that if A is communicating with the B, so on which date paper we have extracted this information. If it's definitely it's ongoing process. Maybe in the future in 2025, A is communicating with C, not the B, because lot of theories are rejected after some times.
Yes, yes.
Means we know, okay, A is communicating V and disease is come into the picture. But in the 2025, they have a new theory, new paper, that they are saying the A is communicating C, now the disease condition, but B communication is not for the disease. So for that, we have made in our disease portal, we always made a portal so that anyone can make an input or suggestion that this interaction is wrong, because we are making this communication, means the directional mapping, we are with our master students, na? So sometimes they make a wrong connection. So we are open to the public domain so that anyone can come and make a comment on this. Okay, this relation is wrong in your network. So we quickly modify the relation and check again the impact. But for the publication, you are 100% right. We always mention in our manuscript, this date we have created the disease map. If any updation or revision in the public available data, we are not responsible, because we are have this data input into the, so we have to always put the date for the safe side, for the communication only. And for the public users, anyone can communicate and just put their input into this, because we are not the expert for every disease, because we are the service provider. We can take input for any disease. We are making the, like if you say, okay, I have requirement for rheumatoid or some other disease, so we take the challenge and we design, but we are not have a full flash knowledge. So we just first design the map and then go to the clinician, go to the domain expert and take the feedback from them, and then give it to you for the publication or whatever.
Okay, that's great. Great.
Yeah. Yeah. Right.
Thank you.
Thank you.
Okay, so I believe we don't have any more questions further. So thank you, Dr. Singh, for this presentation, and it was great listening to you. Moving on, I would now like to invite Dr. Shweta Singh Chauhan, who's a senior scientist at In Silico Minds, and she'll be delivering a talk on From Data to Drug, how AI is shaping the future of medicine. So inviting Dr. Chauhan next.
Yeah, ma'am, I'm sharing the screen right now.
Yeah, it's visible.
Some issue from my side. I think it's a network issue or something.
मनीष कुछ मेल करने गए श्वेता
Now it's visible, ma'am?
Yes, it's visible.
So, good morning, everyone. And it's an absolute pleasure to be here today, especially at this esteemed university where I had a privilege of studying. And I would like to extend my heartfelt gratitude to Prachi ma'am for all her support she gave throughout my journey and inviting me here today. I also sincerely thank to all my faculty members of Amity University for their guidance and encouragement in my BTech and MTech program. So yeah, today I will be discussing a topic that is transforming the pharmaceutical industry from data to drug and how AI is shaping the future of medicine. So over the next 20 or 30 minutes, I will walk you through how artificial intelligence is transforming the drug discovery, the challenges we face, and the future possibilities of AI in healthcare. So as many of you already know that bringing a new drug to a market is a very lengthy process and costly, of course. On average, it takes 10 to 15 years and costs over more than $2 billion. And this process involves several stages, starting from the discovery, preclinical, clinical trials, FDA approval, then post-market. So this process involves several stages such as discovery, where researchers identify potential biological target, such as protein, enzymes, or receptor that play a critical role in disease progression. Scientists screen thousands to millions of the compounds, either by synthetic molecules, natural products, or biologics, biosimilars using high throughput screening and computational drug designing, example, utilizing AI-driven approaches. So overall, the goal of early drug discovery phase is to identify the promising candidates that interact with the targets in the way that could lead a therapeutic effect. Now, after that, preclinical research come into the picture. Once a lead compound is identified, it undergoes extensive preclinical testing in laboratory models, including in vitro, cell-based, in vivo, animal studies, to assess the compound's pharmacokinetic, how the drug is absorbed, distributed, metabolized, and excreted. Along with that, pharmacodynamic of the compounds, the effect of a drug overall in the body and its mechanism of action. In the same way, the toxicity analysis also come into the picture, like to understand what could be the potential side effect and toxicity level at various doses form. The data from preclinical studies basically help and determine whether the compound is safe enough to proceed to the human trials. But however, the problem is that many of the drug fails at the preclinical stage due to compounds' unexpected toxicity or poor bioavailability. So if the drug shows promising results in the preclinical studies, so the companies are basically submit IND, which is called as investigational new drug application, to the regulatory authority. Maybe it's FDA, EMA, TGA, Health Canada, Japan. So basically, this is necessary to begin the human testing and trials to be done. Further, after the application is done and the regulatory is approved to do the clinical trials, then it is conducted in 3 phases: the phase 1, phase 2, and phase 3, where phase 1 is focused towards safety and doses, understanding of the compound. Phase 2 is more focused towards understanding the efficacy and side effect. Phase 3 is focused towards understanding the large scale efficacy and safety in the population patients. And after that, when all the 3 phases the compound is able to cross, then comes the regulatory approval. So overall, the process, the regulatory approval process can take up to 6 to 2 years, during which a regulator basically evaluate the drug efficacy and safety data, the overall manufacturing process, and what kind of a quality measure control have been taken while processing all the phases. then understanding the labeling, doses, and also what could be the potential post-market surveillance requirement, and so on. So after that, when the drug is in the market, then the post-market surveillance is also there. So once the drug approved, it enters into the market, but the evaluation continues, which is called as real world data and real world evidence, which is collected to monitor the long-term safety and the side effects of the drug. So some of the drugs may be withdrawn from the market if there is any kind of a serious concern emerges while the drug is in the market and approved by any of the regulatory body. So despite means the rigorous process and various other consideration, the 90% of the drug candidates fail during the clinical trials due to lack of efficacy, unexpected toxicity, poor pharmacokinetic activities, and so on. Also, it is well known that discovery of a novel drug remains costly, time consuming, and challenging. Everybody knows about all these things. And the pharmaceutical industry is like under pressure to deliver new therapeutics faster and also at the lower cost. And the traditional method of drug discovery are no longer means sufficient to meet the growing demand of innovative therapy. And this is where the AI step in as a game changer, and which offering a solution to accelerate and streamline the drug development process. So of course, artificial intelligence is revolutionizing the pharmaceutical industry. AI can help analyze vast amount of data ranging from genetic information to chemical structure to identify potential drug candidates. This is not only save time, but also reduce the cost significantly. Taking one of the example, like AI can predict how different compounds will interact with biological target, allowing the researchers to focus like on most promising candidate early in the process. So like one of the most groundbreaking example of AI in the field, like one of the company name as Benevolent AI, which basically a pharmaceutical organization have been working on utilizing the machine learning models to identify potential treatment for amyotrophic lateral sclerosis, ALS. And in just 18 months, they have come up with one of the molecules I will show in the upcoming slides. So they are like, they come up with the molecule within 18 months, which basically takes years to do with the traditional method. And this is just one example how AI is accelerating the drug discovery. So AI is not just a buzzword, it's a powerful tool with the potential to transform the drug discovery and development. And this slide overall basically highlights how AI is applied in key areas, utilizing computational modeling, simulation, machine learning to improve the efficiency and reduce the cost in overall cycle. So if we talk about AI in drug design, so AI helps in predicting the 3D structure of target protein, aiding in the structure-based drug designing. It enhances drug protein interaction prediction, allowing researchers to identify potential drug candidates with higher accuracy. Also helpful in drug activity, helping in lead optimization. De novo drug designing uses AI to generate novel molecules focused with specific targets. We already know about AlphaFold, which is developed by DeepMind, which accurately predicting 3D protein structure from amino acid sequences. And the particular AlphaFold is developed by utilizing the AI algorithm, and which is recognized with 2023 Nobel Prize in Chemistry. Another software, if I talk about overall having a various module called as Schrodinger Maestro. So you basically uses AI for drug protein interaction and docking studies along with binding energy calculations. If you talk about AI in polypharmacology, so it helps in designing the biospecific drug molecules targeting multiple pathways in disease like cancer and neurodegenerative diseases. It supports multi-target drug design, improving therapeutic efficacy and reducing the side effect. The same example I want to give, that Benevolent AI is pharmaceutical organization applies AI basically to find multiple target drugs, identifying new treatment for the diseases like ALS, COVID-19. IBM Watson is another company which is more focused towards the drug discovery that uses AI to analyze scientific literature and propose multi-target drug strategies. Now, if we talk about AI in chemical synthesis, it is like one of the major things like in Silico Minds is also working upon, where I am, it's now been 1 year I'm working here. So to understand and analyze the reaction yield, optimizing the chemical synthesis route, it helps in analyzing the retrosynthesis planning, enabling efficient drug manufacturing. Also to understand the reaction mechanism and how we can, reduce the error in the experimentation and reduce the impurity while manufacturing the process. Overall assisting and designing the synthetic pathway and streamlining the drug production. If you talk about AI in drug repurposing, so identifying a new therapeutic target for the existing drug, significantly reducing the development time and cost. It also predicts alternative therapeutic uses and opening new possibilities for known compounds, which is already approved. Incorporation of AI and ML models in the toxicity prediction along with the QSAR, quantitative structure activity relationship models, is helpful in minimizing safety concern early in the development. Also helpful in enhancing the bioactivity prediction, increasing the likelihood of finding effective drugs. Along with that, the drug likeness property analysis, physicochemical property, pharmacokinetic parameter, which is very much crucial for drug formulation. So all these things are like the examples where AI can help support in the process of drug development. So this slide basically outlined the role of modeling and simulation in drug development. As I have already talked about, overall AI can help in any of the end-to-end cycle. It is spanned from discovery, either by drug repurposing, target identification, bioactive prediction, to non-clinical QSAR, ADME and toxicity analysis, and in the clinical phases to understand the virtual patient simulation, adaptive trial design, meta-analysis to optimize the drug development process. So here are some of the few examples where the computational approaches that enhance the efficiency in identifying and optimizing the new therapeutics, or drug repurposing, where AI can analyze existing drug to find new therapeutic application. Along with that, target identification, as in the previous lecture we have already gone through with the structural biology approaches to identify the specific target and further move towards the docking and MD simulation. So this is one of the major role of bioinformatics. Like it's one of the crucial application where AI helps in identifying potential molecular target for drug by analyzing the biological data, which is already present. Another focus is bioactivity prediction. So by use of machine learning models to estimate how the compound will interact with the biological system, along with the similarity, or based upon the similarity of the compounds, and scaffold analysis, helpful in identifying the bioactivity of the compounds. Molecular docking, everybody knows that further refines the process by simulating how small molecules can bind to their target proteins, which is essential for structure-based drug designing. In silico toxicology is also play a very important role in predicting the compound potential toxicity using computational model, helping in minimizing the safety risk early in the development. Similarly, ADME predictions provide insight into the drug pharmacokinetic, ensure better efficacy and safety profile. Along with that, safety and efficacy prediction models are also there. So they can basically identify what based upon the structural similarity and matches the similarity with the tanimotor coefficient or the scaffold, which and understand like what kind of a fragment or the functional group is responsible for causing any kind of a toxicity. Either it is mutagenicity, carcinogenicity, developmental toxicity, ocular irritancy, or ocular sensitization altogether. And that can helpful in identifying there are various software and tools are available to identify how the particular molecules can give the adverse effect and can be analyzed early in the drug developmental stage. So if we talk about, this is all about the early drug discovery. And if we talk about beyond discovery, AI also improves clinical and manufacturing process. So this is about dose optimization. So there are models to determine the most effective and safe doses based on patient data. Disease modeling is also one of the major applications where the simulation of the disease progression is done to study the treatment effect of a drug overall in silico. Trial design optimization also is there to ensure the efficient, cost-effective clinical trial by predicting the potential outcome. Then real-world evidence and real-world data is also like very much important to support the regulatory decision and post-market surveillance. Coming to the picture, virtual patient and digital twins, where now most of the industries are more focused towards. So that here what is done, like simulation of human responses to the treatment is done to understand to improve the drug testing overall and reducing the reliance on the animal model. So this is lastly, if we talk about AI enhances also helpful in manufacturing process by optimizing the drug production, ensuring the quality and scalability. So if you see here, it's some of the few examples of AI in actions. So the companies like in silico medicine, Atomwise are basically using AI to identify new drug candidate for disease like fibrosis, Ebola. In one of the case, AI discovered drug candidates for fibrosis is now in the clinical trial. If you see here, like Recursion, a small molecule for pan-SGAC inhibitor, it's in the phase 2 and 3. Similarly, these are some of the major organizations worldwide. They're working on utilizing AI and ML techniques to identify the ligand and the compound molecule against the specific diseases. So another example, if you see Extentia, which is used AI to design a novel molecule for treating, obsessive compulsive disorder, OCD, in just in 12 minutes. Basically, a process typically takes 4 to 5 years. So this is just example that shows how AI is just not a theoretical term, but the people are started applying it, and it's making a tangible difference in fighting against different diseases too. So this is like another, just I think on 24th March, this news came that Rentuserib is a new drug which is developed by in silico medicine to treat the idiopathic pulmonary fibrosis. And it's basically a serious lung disease. So so they have discovered both the drug and its target from utilizing the artificial intelligence and ML models, and that making first of its kind. So this success also shows that how AI could change the way we develop treatment for many diseases in the future. Now coming to the regulatory agencies such as FDA, how they are utilizing and seeing and adapting the artificial intelligence and machine learning to process. So here is like I have taken the particular presentation slide from the FDA itself, and they are very much focused to utilize the artificial intelligence and machine learning model to support and analyze the model building during the submission of the drugs for the approval, if we see here. Similarly, if you talk about major regulatory bodies, FDA, of course, they oversees drug approval and regulation in US and overall in the world. They are providing guidelines on clinical trial, good manufacturing practices, pharmacovigilance, and utilization of AI/ML application in drug discovery. So FDA, along with ICH and Center for Drug Evaluation and Research Division, regulate new drug applications for biologics and along with biologics and generics drug development. Similarly, if you talk about EMA, they are also like regulates the drug approval in European Union. It follows ICH guideline and has a strong focus on real world evidence and digital health application. So the EMA that basically support the 3 R's: replacement, reduction, and refinement. to limit the animal testing and promote in silico modeling. Another example, if we talk about the CDSCO, which is Central Drug Standard Control Organization of India, which is an Indian regulatory authority for pharmaceuticals, and it aligns with ICH and World Health Organization (WHO) guideline for global harmonizations. So they are more focused towards approved drug and overseas clinical practices, and regulates how AI-driven tools can be incorporated into the drug discovery life cycles. Now, International Council of Harmonization (ICH) is basically regulatory guideline which is adopted by many of the regulators, either it's FDA, EMA, CDSCO, PMDA, Health Canada, and others. They are more focused towards the quality, safety, efficacy, and utilizing multidisciplinary approaches. Quality, it means focus towards manufacturing, impurity, and stability testing. Safety is toxicology and non-clinical studies. Efficacy is more focused toward clinical trial and benefit risk assessment. And multidisciplinary is utilization of AI ML application and pharmacovigilance. So the market of AI in drug discovery is like growing very rapidly, and it is estimated by 2032 it is projected to reach $14.5 billion, which is like a compound annual growth rate of 20.8%. And this growth is driven by increasing the adoption of AI solution across pharmaceutical industry. From early age research to clinical trials, companies like Benevolent AI and Silico Medicine, Recursion Pharmaceuticals are leading in charge, demonstrating the potential use of AI in revolutionizing the drug development. So despite various potential applications and everything, AI in drug discovery is not without challenges. So data quality and availability remain significant hurdles. Also, there is a need of more robust validation of AI models, which is very much necessary to ensure the predictions are accurate. Along with that, integration of AI into existing workflow can be a complex process and requiring significant investment, infrastructure, and training of individual. However, if you talk about the companies and the industries are more focused towards utilization of AI in drug development process, shows that the challenges can be overcome when there is a right approach and collaboration between academia, industry, and regulators are done in promising way. Further, the future of AI in drug discovery is incredibly, promising, that revolutionizing the multiple aspect of drug development process, and advancement in AI model will enhance target validation, increasing the likelihood of success by focusing on most promising target. And if you talk about the personalized medicine, it is helpful in treatment of individual genetic profile, as we have seen in silico medicine efforts in developing customized cancer therapies. AI is now advancing in predictive toxicology, identifying potential side effect early in minimizing the late stage failure in the preclinical and clinical trials, such as Recursor Pharmaceuticals, for instance, now utilizes AI to predict drug toxicity and reducing the risk of optimizing safety. The clinical trials are also now become a more efficient, way with AI-powered predictive analytics, helpful in streamline the patient recruitment process and improving the retention rate. So overall, AI ability to personalize treatment, predicting the side effect, optimizing the clinical trial is reshaping the future of drug discovery and healthcare system altogether. So yes, AI is transforming the field of drugs discovery and development. So either starting from the accelerating the target identification to optimizing the clinical trial, it has a potential to transform the way we develop new medicine. The success companies like in silico medicine, Benevolent AI, Recursive Pharmaceutical, demonstrate the power of AI in solving most of the complex problem in the current scenario. So of course, challenges remains in every field, but the future of AI in healthcare is incredibly promising. So this is all from my side today. And thank you, everyone. And I'm grateful for this opportunity to share the insight with you all. And I would like to thank you to all the faculty members along with Prachi ma'am, and to the audience too. If anybody have any question or would like to discuss further, and I'm happy to engage. Thank you once again, everyone, for your time and attention. Thank you.
Very nice talk, Shweta. Thank
Thank
you.
you so much, Shweta, for nice talk and nice presentation on very important topic. And now you have given really in-depth understanding of this subject. So, dear audience, if you have any questions or any query, then you can ask.
Ma'am, I have a question.
Yes.
Ma'am, what are the biggest challenges in integrating AI into pharmaceutical research and development?
So if we talk about the challenges like I have already talked about, like one is the major challenge is the data, because everything is dependent on the data. If there is a saying that if you're putting the garbage will come as a result. So it's very necessary to analyze what you are, how you are doing and developing a model, what kind of a training set you are taking into consideration. And of course, the validation process is very much necessary to take in consideration.
Okay, thank you, ma'am. Ma'am, I have another question also.
Yeah.
Ma'am, what are the regulatory and ethical consideration when using AI in drug development?
Yeah, if you talk about if you talk about FDA, EMA, so in Silico Minds, where I'm working, is more focused towards the generic drug development and how we can support, um, how in silico support in the generic pharmaceutical development. So if you see here, various guidelines are given. If you talk about ICH, so they have provided M7 guideline where whether you can utilize they have mentioned that you can utilize the in silico models for the prediction of mutagenic analysis, but you have to provide the background and each and every information from what kind of a tool you have uses, what the backend the tool is working upon, along with the validation based upon the control molecule you have selected. So they are now the regulatory bodies are more focused towards adapting the use of AI and ML in overall drug discovery process, either it's generic, biosimilar, or biologics, or early stage drug development. But it's depend on the product to product how the challenges are there. But for the generic drug development, if you talk about all the regulatory bodies, they are more accepting and they are bio-waiving the clinical trials too. Sometimes phase 3 clinical trial, somewhere I have heard that phase 3 or phase 2 clinical trial for submitting the generic drug development, generic drugs.
Thank you, ma'am.
Thank you.
Okay, Shweta, thank you so much. Anyone else who has query?
I have written one question in chat box.
I also have one
request
question.
chair.
I also have one question.
I request to chair, please proceed my question.
Dear Dr. Shweta, I also have one query. How AI is helpful? How AI is helpful in improving diagnostic accuracy and reducing errors in medical practice?
And now it's like what I'm seeing in my organization, because we are not more focused towards the drug device and combinations, but itself like now AI and some of the ML programs are people are utilizing to understand what could be the size of the injections and how, if it is inserted in the skin, how much the particle size is distributed, at what force, and at what particular and the diameter should be of a needle should be there and everything. So now the people are more focused towards that and utilizing the use of AI and ML in this field. I know about the drug device combinations that utilization of not to properly say AI and ML, but some kind of traditional computational approaches that people are more utilizing.
Okay. Thank you so much. Now, Prachi ma'am.
I request to chair with due permission. Shweta, very nice presentation, and definitely everybody is talking about AI. So we are not bounded with the AI. We are most of the manual. So can you give any example of AI driven drugs, those who are being designed in the current time?
Yes, ma'am. Of course, I have just shown the list also, I think, of the drugs. Such as if you talk about recently, the one of the drug name is rentosertib, basically, rentosertib, which is designed by in silico medicine. And against the IPF is idiopathic pulmonary fibrosis. And so they have utilized, they are saying that they have utilized and discovered it by utilizing the AI and ML based models, both the target identification along with the ligand generation, the compound identification. Both they have done by utilizing they are complementing the traditional computational aided drug design by utilizing some of the models in between the process of identification.
Okay.
Along with that,
Thank
there
you
are
for
various
updating
other organizations. If you talk about, there is one more pharmaceutical organization which is called as Recursion Pharma. Then there is another one is Benevolent AI. They are now in the clinical phases, and with the drugs they have developed and identified by utilizing the models which are based on deep learning neural network, and the backend the AI models are utilized. Of course, the traditional thing need to be there, the manual thing need to be there, but how we can, if there is a 10,000 molecules and there's a 1 million of molecules need to be screened out, then how we can incorporate these approaches at the back end.
Thank you, Shweta, for updating.
Thank you so much, ma'am, for
nice
this opportunity.
Yes.
Thank you, Jyoti, ma'am.
Thank you so much, Shweta, for nice presentation.
Thank you so much, ma'am. It's a pleasure to be here and meet you all. Thank you so much. Thank you, ma'am.
Now I have one more speaker. I would like to invite Dr. Jitendra Narayan, Senior Bioinformatics Scientist, CSIR, IGIB, Government of India. Title of the talk is Comparative Genomics of SARS-CoV-2 Evaluation, Adaptation, and Genomic Insights. Dr. Jitendra Narayan, please share your PPT.
Yeah, hi. Good morning, everyone. I will share my screen to. Am I audible to everyone?
Yes, now you're audible.
Just wait, give me a second. My screen visible to everyone?
Yes, it's visible.
Is this visible to everyone?
Yes, it's visible.
So, hi, good. Thank you for nice introduction. So, myself, Jitendra Narayan, and I am a computational biologist. More specifically, I'm trained into comparative genome analysis. And I am today going to discuss comparative genome analysis of SARS-CoV-2, the evolution point of view, adaptation point of view, and genomic insights. So before going deep into SARS-CoV-2, I wanted to start with the genomic surveillance first. So let's talk about the genomic surveillance. So when we talk about the genomic surveillance, we instantly remember only one test, RT-PCR test that we use almost every day during the surveillance time. But apart from that one, we also have protein-based detection being used in genomic in COVID surveillance. Antibody based test is also, like serological test, is also being done during the COVID times. But the one thing that is really interesting, really useful for us is genomic-based analysis. So for my domain, it's genome-based analysis, and it does have a lot more information than just identifying that we do have viral positive or negative. We also have more information like genomic information that we can use and identify the specific mutation that is actually occurring in a certain population, and they're more likely the next consequences. So so the one thing that actually attracted me is the genomic-based diagnostic test. And we started working on whole genome data analysis of SARS-CoV-2 and try to understand why and how these viruses are evolving over the time. So genomics sequencing has a really crucial role in understanding the genomic variation of the viruses. Most of the time, we do have COVID positive or negative, but we were not able to identify that certain mutation is actually occurring in certain population that we can actually pinpoint in using genomics data. Apart from that one, we can also use the genomic sequences to monitor just the evolution of the virus. So overall, how does virus is evolving? We can monitor with the help of whole genome sequencing data. Apart from that one, we can also pinpoint and keep an eye on circulating COVID variants using whole genomics data. So out of all these clinical surveillance does have many advantages and disadvantages. Like if you look at, just give me a second. Okay, so if you look at clinical surveillance, the sampling biases is there. Most of the time, clinical sensitivity is also an issue with clinical surveillance and time and resource constraint. So we also have, if someone is doing the clinical surveillance, they have time and resource constraint. And the most important thing is the ethical and privacy consideration. So if someone is doing especially the COVID surveillance, doing the COVID surveillance, we also face that some people don't want to share the data. Some people don't want to share the data that we are infected with certain, let's say, variant. So they were not very happy to share that data. So these are actually stopping us to do the clinical surveillance. We cannot go and reach out to each and every person in India and say, hey, I want to do the surveillance and I need your sample. And they are not very happy because they don't want to share that what is the age, what is their location, and many more things that they don't want to share. They hide these information. So yes, they do have some limitation. But if you look at the other hand, we do have another surveillance system that is widely used nowadays. Most of the research organization, especially the surveillancing sectors, is focusing more on wastewater data. So as I mentioned earlier, clinical data has many limitations that we certainly need to take care of when we are doing any kind of surveillance. But if you look at the wastewater data, it has many benefits, especially the early detection of outbreaks. So using the wastewater data, you can even predict the possible evolving variant in that society, in community, or in that country. You can use that outbreak early detection methods using wastewater data. That is the one beautiful thing that we noticed, and the people instantly started switching towards the wastewater data because we do have a lot more information, and we can more likely predict the possible evolving variant. Community-based surveillance, we can do surveillance using the wastewater data. What is happening in, let's say, Gomti Nagar, for example? So we can tell them using the wastewater data surveillance analysis and what is going on in that community, kya ho raha hai, we can tell them all together. Because the beauty with wastewater data is that's really, for example, you are doing kind of surveillance, you can not only identifying specific viruses like COVID, but you can also have many other viruses sequenced in one go, and you can do the analysis work. You are not specialized in growing a specific let's say, strain and then sequencing it. You are having the detailed idea that we also have, let's say, Omicron, Delta, and all the variants in one go. So, actually pinpointing the specific reason. Third one, the next one is the tracing. So you can, that's really important for COVID, during the COVID surveillance, that we need to trace back where it happened for the first time, where these Omicron variants appear for the first time, who is actually transmitting these viruses. So this kind of information can be easily answered using wastewater data. And multi-pathogens, so not only one pathogens, we can have multiple pathogens in one go if you are dealing with the wastewater data. So these are the beautiful things about the wastewater data. And we quickly switch to the wastewater data analysis in my lab and many other collaborators as well. So what is going on across the globe now? So most of the time, you rarely nowadays will hear that we have a clinical surveillance or something. We have mostly the wastewater surveillance. People are silently doing the surveillance just using the wastewater data. So this is a water scan dashboard. So they are actually doing all these scanning across the USA and trying to pinpoint a specific new variant evolving in that region. the similar fashion, there is COVID-19. So they are actually studying the tools of the specific location and trying to identify the infected individual and their possible new evolving variant. So, and the interesting thing about this surveillance dashboard is that the many people, especially 289 universities, are connected with this network, and they are actually submitting the data and submitting all this information to this dashboard so that we can easily trace back all these evolving variants across the globe. COVID provides many other information, for example, universities, and if you look at the country point of views, we have 72 countries that are actively contributing into it as well. So in India, I am actually my lab and CSIR is actually CSIR IGM is actually involved into wastewater surveillance, and we are mostly I am actually mostly my lab is actually associated with the let's say metro city surveillance, where we are actually doing the metro city surveillance, and the consortium name is APSI, and we are doing the surveillance of Bengaluru, Maharashtra, and Delhi. All these regions are being surveilled during the COVID times, and we can trace back all these new evolving variants using our dashboard as well. So any tools that we can use, I mean, if someone is into let's say COVID data analysis, they are mostly familiar with this. So Fraser is one tool that report the relative lineage abundance. So it's really old tool, and nowadays most of the people prefer to use Fraser. Callisto is one RNA-seq tool, and this is being modified just for the metagenome transcriptome data analysis. When LCS is the lineage decomposition SARS-CoV-2 pool sample data analysis, whereas Kraken is also is also a well-known tool amongst the bioinformaticians. And this is being modified for the COVID and data analysis as well. CWAP and Aquascope is another pipeline for lineage estimation. And there is Lollipop, LCOP, and lineage spots. These are the other pipelines that is being developed to just study the abundance of specific lineages for COVID. So if you remember the COVID, initially we had only few lineage, let's say strains, variants at that time, but now we have a lot more. But interestingly, if you pinpoint a specific variant and notice, so you can see here, BA2 is actually evolving multi-directional. So let's say BA2 is evolving to BA2.35, BA2 is evolving to BA2.3.221. So they are evolving differently. They do have a different mutations altogether. But if you look at pinpoint here, so BA2.75 evolved to BA2.7.9, and then they evolved to CB.1. And CB.1 has B.1.4.1. They evolved to XBB4. And XBB4 and CH.1 is actually resistant to Betello remote. So these are getting resistance, and it is not only one single part. So multiple strains are getting resistant to all these variants, and they are doing the convergent evolution. So this is one clear example of convergent evolution where two different lineages evolving differently, and they're getting resistant to the similar kind of directions. Okay, so I mean, it does have many implications if someone is doing the whole genome data analysis. So convergent evolution can have many information that have some challenges as well. For example, misinformation. It might be possible that we misidentify a special variant. That is one challenge. The mutational portion. So which portion to do the analysis is also a concern if someone is doing the convergent evolution analysis. I'm sorry. Biases, sampling biases do occur if someone is doing this kind of analysis. And then the methodology and pipeline. So we do have a limited methodologies and pipeline, and we still need certain improved and advanced pipeline to do this convergent evolution study. So let's talk about the region first. So most of the time, people do focus on a specific region. Let's say key surface protein, for example. So mutational portions analysis a portion of the mutation of interest rather than lineages. So people do not study the lineage, but they only study the specific region of the genome that is of important for them. And that you can do as well. So my lab is more into designing new software and new pipelines as well. So we design, I'm sorry. So we designed CUPI. CUPI is a knowledge-based understanding of pathogen invasion. So the use of CUPI is to unleash the power of ancestries to unearth novel variant and beyond. So we are trying to identify the possible evolving novel variant using CUPI pipeline that we develop in our lab. And it also report and classify the unknown contigs, unitics, and detect the emerging variants. So the idea was to identify the emerging variant and unearth a novel variant that we identify in wastewater data or during the clinical surveillance. So what is and how does the CUPI work? The CUPI work is kind of straightforward, but we have some new approaches, innovative approaches to deal with the evolving variant. For example, if you look at here, the input file. So input file is the FASTQ file that we receive from the sequencing data. Then we remove and do the using thermometrics, we remove the adapters, and then we filter using SYMPOSIUM. Then using the quality control, quality checking, we take the best quality data and check for the possible contamination in it. And then we map against the reference using the BOTAI, and we created a BAM file. Using a BAM file, we created a D1 graph of this entire data set. So the beauty with this D1 graph data is that we do have a unique tick. So unique tick and contigs. So most of you are familiar with contigs, FASTA, and blah, blah, blah. But unique ticks is a unique contigs. So always remember, contigs is contiguous sequences, whereas unique tick is unique contigs. And then unique ticks is being sent to SEQ-Kit pipeline to where they locate specific unit tick into the GSAT database. And if it is mapping to GSAT database, we call it, okay, this kind of sequences is known and submitted to GSAT, so it's not useful. If it is not mapping to the GSAT database, then we extract them and we further try to look for using BBMap, try to extend the possible unit tick and form a contiguous sequences. And then we use the next clade. So the next is the next clade where we use the clade mutation and lineage assignment. So we assign the specific lineage using next clade, and we had DC, mutation, coverage, and KMR. So we had many information. We reported DC percentage, what is the mutation, what is their coverage, and KMR, and further did the scoring using all these information to tell them that we have possible evolving variant. So using the same data that we received from all these metro cities in India, I took the only data from the specific time points. Is November data. I took it from the clinical samples. And when we look into it, we were able to identify the possible unitics, and that has the unique mutation in the genome. When I compare the clinical samples with the CUPI data analysis approaches, we were able to report that even in the November time, we were able to report them that B.1 is in the data set. We do have a recombinant XB in the data set that was there in the November data sample. We're using wastewater data. So it was not reported using the clinical samples, but in wastewater data, we were able to identify XBB and XB. These are the recombinant one that we were able to identify using CUPI data analysis. We further look into the possible new mutants, and we noticed that the new mutants started evolving during the November time, and we had new evolving variants. So the CUPI advantage. So what is the advantage of CUPI? So that's they identify the novel fragment that is not known to SARS-CoV-2 and wastewater sequencing data. So we can identify the novel fragments in the wastewater data and report them. Trace back the evolutionary variants. So we can trace back the possible evolving variants in SARS-CoV-2 lineage. And early detection, so we can also identify early detection of lineage before they gain dominance. So we can even report them the possible evolving variant that might get dominant over the time. Apart from this copy, my lab is in silico bioinformatics data analysis. So we noticed that most of the time people do have a specific problem at doing this genome assembly as well. Most of the data that we have in the resources in the public repository are not having good assembly, and they are actually fragmented, or they have a lot more and in the genomic sequences itself. So we decided to have our own tool that can do this quality control, genome assembly, and reporting the best quality genome sequences using SARS-CoV-2 data. So these are the pipeline here. So the read files, we took the read file, we do all these QC tests where we coverage check, adapter removal, and read length filtration. And then further this BWA and coverage plot analysis, and BAM formation. And then we had using SPADE and RegOut, we did the assembly, and we filled the gap using RegOut and gap to SIG pipeline, and had the best possible assembly. We published it into microbiology resource announcement, and it's freely available. If you are interested, you can reach out to my GitHub page where you can download and play around with your scripts as well. So when I compare the SETU pipeline with the existing pipeline that we have for SARS-CoV-2 data analysis, we notice that we do have genome fragmentation, and better N50, longer fragments, longer contigs, better N50, and the time is even better than the rest of the pipeline. So relatively, SETU is doing better, and we do have better performance for using all these parameters, and is being reported to the journal. The third one that we are actually working nowadays, and we are actively improving it, is BHEDI, BHEDI. So it's actually biomarker-based heuristic engine for dengue identification. So as I mentioned that SARS-CoV-2 is there, but in addition to that one, we can also identify other viruses in the wastewater data as well. So I was really curious to know, can we identify dengue as well in the same data set that we are receiving for the during the surveillance? So we started looking into the specific dengue wastewater data sequencing, and then the read quality, and then we applied the complete pipeline, the workflow here. So we took all these serotypes, serotype 1, 2, 3, and 4 of dengue, and created a KEMER set and the SSR marker set. And we both combined together and form a possible markers that can easily identify all serotypes, serotype 1 to 4, using our pipeline. And people can use the dashboard to visualize the data and interpret it online as well. So it's freely available. If you want to, you can reach out to my website, web server, where people can reach out and further discuss this as well, and further explore this as well. So this is one interesting pipeline that is still under publication and under communication. So we'll update you once it is published and freely available to the public. Okay, now we do the comparative genomics section that we are actually doing, and we recently published our tool on comparative data. So what we did is, okay, so it's normal that people are doing the sequencing. We are getting the sequencing data. We are doing the genome sequence assembly and submitting the data onto the server. That's all. Is this what we are looking for? No, we are perhaps looking for the possible in-depth knowledge from that data. And so what we did, my research lab did is that all the sample from India, so around 2 lakh samples from India, we do the QC, and we end up into having 64,000 best quality genome sequences that we can use to further do the comparative data analysis. So we divided the data into 3 sets. So the age, based on the age, we divided into 3 sets. early age when people 1 to 17. So if the age is 1 to 17, then is children. Then if it is 18 to 64 is adult. And if it is elderly or age above 40, 65, we call them elderly. So we have children's working group and an old age people. In the same time, vaccination, we have say what happened before vaccination, after vaccination in COVID data, to look into the COVID data. We also look into the geographical states. So 25 states and all territories, like we try to look for the geographical states, what is happening in there. And we focus on lineages, unique substitution, and mutation frequency in the data set. So this is all the idea to study the 64,000 genomes from India to understand what happened to each vaccination and geographical region for SARS-CoV-2. So we noticed when we started looking into it, we noticed that in comparative genomics data analysis, most of the data that we are receiving and coming from and the unique mutation that we are looking into is actually from Maharashtra. Maharashtra is the topmost where we do have a higher number of unique mutations occurring in the genome. Apart from that one, the Andhra Pradesh and other states are mostly the southern states are mostly have the unique mutation as compared to the northern states. When we look at the comparative genomics data, we were surprised to know that once we divide the data into 3 sets, so before vaccination, or during vaccination, and after vaccination, and once we noticed that most of the unique mutation that is occurring in the viruses are actually getting increased after the vaccination. And we have decided that more likely, it might more likely possible is that we are getting more resistant towards after the vaccination, and we are stressing the viruses to create more mutation, and this is the more likely reason that they after vaccination they have increased the number of unique mutation before and after the vaccination. We were also interested to know that in all 64,000 genomes, do we have any specific region in the genome that is undergoing mutations? And we noticed, yes, we do have specific. So old age group have a different mutational pattern as compared to the middle working class group as well. So they have altogether different mutational pattern across the genome. So thank you for your time. Thank you. I'm really sorry for my voice because I do have a cold and I am coughing as well. So I'm sorry for that one. So that's all. Thank you.
Thank you. Thank you so much, Dr. Jitin, for delivering such an insightful and thought-provoking talk. And your valuable perspectives and in-depth knowledge greatly enriched the session. So once again, thank you so much for nice presentation, nice talk on very important topic. Dear audience and participants, if you have any questions or any query, ask. Expert is here.
Dr. Jitendra, I would like to compliment a very informative talk. And being the organizer, I cannot concentrate 100%. Okay, to be honest, at my part, but the session was very good and it was quite informative. It is my very genuine and humble request. Can you display any demo type of thing? What the pipelines you have designed, if you have time or if you have any availability, so that students can be benefited to see the real-time application of bioinformatics pipeline, how you scientists are using during the time of COVID to fight against the COVID, how you people like you have designed so many pipelines, it is on the GitHubs and many other places. So if your official things are permitting, so can you share with our students?
Screens are visible to you, my tab. It is visible? No? So I will share my entire screen and then I will discuss with you. So just give me a second.
Yeah. Because they can get certain idea about the hands-on, how they use these kind of a single demo only, not much, a single demo. Yes.
It's visible to you? my screen is visible to everyone, right?
Yes.
Yeah.
Yes, it is
So,
visible.
yeah. So what actually I notice is that most of the time, whenever we do any kind of enrichment analysis, I am pretty sure that every one of you know some enrichment tools for sure. For example, David, for example, Cynigo, David is the most popular one that most of you use for the data analysis, or for especially the enrichment analysis. I noticed that we do have a lot more tools online, but the problem is if you are working on human genome mouse models, or let's say Drosophila or C. elegans, you are lucky because you do have a lot more information and tools online that we can use. But if you're working with something, let's say some unusual bird or some unusual fungus that is not known yet, then there is a problem on how to do the enrichment analysis. For that one, we and my lab actually developed a new tool, and we call it Get Enrich, where it can automatically do the enrichment analysis of all non-model organisms. When we call non-model organism, I'm not talking about C. elegans or Drosophila or zebrafish, or let's say other mouse models. But the things that is completely unknown, for the first time someone is doing the sequencing, and we are interested into knowing the specific pathways and pipelines to figure it out. How does it work and which pathway is enriched? For that one, you can use Get Enrich. So overall flowchart. So we do have a complete flowchart where you can use BLAST Coila, EGG Nog, and CAS. So 3 softwares we do have dependency. So either you can use BLAST Coila or ENOG or CAS. We need all outside from all these software and process this, and then from a process annotation file. And these annotation files are used by Get Enrich. Get Enrich further with the KEGG annotation, and then subset data analysis. And further, they go with the pathway enrichment analysis, where we use the KEGG database in the background, and further do the clustering analysis. And then we do the enrichment analysis and report it to the public. So you will have a lot more data. If you are, let's say, for example, running Get Enrich, you will have a bar plot and all these kind of plots using one click. So you just need to upload your genomic sequences file, let's say in faster format, and you will have the complete Get Enrich plots, all these plots. So we have plots, we have adjusted p-adjusted value as well. So all these plots is available once you run Get Enrich. You can also have the path view, and in path view we have two different path view adjust.r and path view p-value.r. These two packages can be used to further draw a PNG for further analysis work as well. So, and apart from this one, we also track this software directly, track all these PubMed databases and try to look for the specific pattern that is being submitted to PubMed databases and plot them as well. So you can also have a PubMed trend plot using this Get Enrich tool. So the beauty with this Get Enrich is that you don't need to write even a single line of code. You only need to provide 3 files that we required, and then you're going to have the complete data analysis done and all the plots generated for you as well. So these are the plots here. Okay. So we do have a GitHub repository as well. So if you are familiar with GitHub and programming, you can also go to the GitHub and you can do it from the scratch using your web server. If you don't know this, you can click on the start analysis and this will start. So these are the real interface for the data summation. For example, if you are into, once you click on this analysis, so this is the interface where you have Get Enrich and file formatter. So you can run the Get Enrich, or we have Get file formatter. So you can click on this one and you can change the specific file format that you need. So we do have everything online. So you can upload the file, pass output file, coil output file, and eggnog file. and it processes it. You can click on Get Enrich, and then you'll have, you click on Get Enrich, then you'll have a Get Enrich page where by now 314 job is being submitted on Get Enrich web server. And the last day that the job is submitted on the web server is this, and the number of job in the queue. So 5 jobs are still running on our server as well. So here is the specific setting. So once you do the enrichment analysis, you need to provide your email ID here and all these background genes, gene of interest, and the KEGG annotation file. So you can click on the browse file and you can upload the specific background gene, gene of interest, and KEGG annotation file over here. And you can also adjust your organism code. So we noticed that most of the time, once people are working on, let's say, Drosophila or C. elegans, they do have, they know their genetic code and everything. So if you know your KEGG ID, so you can provide your KEGG ID here, for example, KO or something, some ID that you know. So you can write it down here as well. You can do p-value and p-adjusted threshold. So you can set the threshold. For example, by default is 0.05, and you can do the further setting, and you can change this threshold as well. So if you are interested in 2.10, so you can change and run the job. Apart from this one, we by default, it's the default one that we're running. If you are into p-value focus and try to focus on p-value based significant pathways, you can click on this one and just focus, prioritize just p-value significant pathways and comprehensive. So all these are available here. So we have 2 options in the pipeline. So p-values, so if you have generate heat map, for me, generate this one and click run, and it will run it for you. And then you can also select the one plot that you like the most, and you can run, and you can click on the start analysis, and it will run on my server. And once the job is complete, your result will be on will be available to the check result. So once you click on the check result, you can provide the ID that is being provided during once you submit the job. They'll provide your ID. We'll provide you with ID, and you need to enter your ID over here. And then once you click here, you'll have all the detailed job status, the running status, and you can also view the result as well. So you need to provide the ID. So once you submit your job, we'll create an ID, a unique ID for you. That unique ID will be used here to see the results. For enrichment, so you can do all these enrichment analysis, p-value and p-adjusted, and all these data will be visible once your job is completed. So the basic instruction is written here in the left panel. You see here the basic instruction, okay, what to do and how to run is here. So if you need further information, you can go to the user manual. So user manual have all these detailed one step by step information. So you can use this user manual for during the submission of your job. Apart from this one, we also provided some sample files for you. So for example, is the sample file for human genes. So you can download the background genes, gene of interest from here, the sample file, just to understand the file types that we need to run this get Enrich program. KEGG annotation files. So how does the KEGG annotation look like? Background KEGG annotation, background gene of interest, background KEGG annotation, all these information. You click here, it will download it for you, one sample file that is needed, and you can check your input file format for you. You can also use the force and permit trend by clicking here in the box. So that's all. I'm really sorry. So that's all, and you can use this one. And I will love to hear from you and to see the updates on Get Enrich. We are actively improving this web server so that it will be useful for the broader audience as well. So that's all for today. Thank you.
Okay.
Back to you, ma'am.
Nice demonstration. Jitendra, students have one or two questions, Dr. Jitendra.
Mhm.
So please.
Excuse me, sir, I have a question.
Go ahead. Go ahead.
So is this pipeline freely available or we have
It's
to
freely available.
purchase
It's
it?
published now. Okay, so that's a good question actually. So most of the time, most of the people do prefer to have open source stuff, and I believe in open source. So this entire code is available on GitHub. If you are familiar with the GitHub codes and softwares, installation, everything, you can clone the code GitHub and run it by yourself. If you are not familiar with this, you can go to the web address getenrich.igib.res.in. It's written at the top here. You see my screen here. I'm selecting this. So getenrich.igib.res.in. You can go there and directly run it. You can, even if you try right now, you can see the specific interface that is running in my system as well. So it's freely available and published in Bioinformatics Advances, and it's freely available to the globe.
Okay, so thank you.
Anybody else? Any question? Am I audible to everyone?
Yes, you are audible, Dr. Jiten.
So any question or any concern that I'll be very happy to answer. And if there's no any question, then anyone can go ahead and handle this.
Sir, I have a question.
Uh-huh.
What are the key genomic differences between SARS-CoV-2 and the other coronaviruses? Like
Okay,
SARS-CoV-1
we do have a
was...
mutation pattern. So if you remember my slide, so here, so just wait a second. So if you look at my slides here, okay, just wait a second. So if you look at the slide here, so we do have a lot of new mutations. For example, B.2 has a mutation at 444, and they're becoming B2.2.3.1 and 2. So they have a mutation in at 346 R is converting to T and forming BA.2.76. We do have a specific mutation that define that lineage. Okay, and sometimes there is only one mutation that completely define that lineage. For example, here say B.1 is actually R346K and then forming BA.1.1. So they have an evolved variant to BA.1.1 and only after having this mutation. Yeah. Anybody else?
Any other questions, audience? Okay, Dr. Jiten, thank you so much. Once again, thank you so much. And I'm also very thankful to all the speakers in this session. First, Dr. Krishnapal Singh, title was Role of System Biology in Target Identification Using Disease Maps. And our second speaker was Dr. Shweta Singh Chauhan, and topic was from data to drug, how AI is shaping the future of medicine. And the third talk was Dr. Jiten Narayan. Title was Comparative Genomics of SARS-CoV-2 Evaluation, Adaptation, and Genomic Insights. So all three lectures was well structured and highly informative session. I think that enriched knowledge and understanding for everyone. So once again, congratulations and thanks to all the speakers. Thank you so much. And now I would like to request organizers of this symposium, please make an announcement for the after lunch session.
Thank you so much, ma'am. Now we will have a short lunch break to energize ourselves and come back with more enthusiasm and energy. The session will reconvene at 1:50 p.m. Our chairperson. I thank our chairpersons, Dr. Jyoti Prakash ma'am and Dr. Nikita Basan ma'am, and the rapporteurs.
Thank you so much.
I extend very warm thanks to my chairpersons, Dr. Jyoti Prakash and Dr. Nikita, and very special thanks to our rapporteur of the session. They are doing their job wonderful. So really, the session you made very nice. Thank you so much, rapporteur, for this. Garima, our reporter of the session, Dr. Osaf Ahmed and Dr. Rachna Singh. Thank you for joining and supporting the organizers for conduction of the symposium. Dr. Jyoti Prakash and Dr. Nikita ma'am, thank you so much for sharing the session so smoothly.
Welcome, Prachi ma'am. Thank you so much.
Thank you to our reporters.
Just a minute, the point will be detected if the participant are not going to show their face, right? Because this is your oral presentation and you need to be here, in the online, your presence has to be online, right? So no excuses will be allotted to the person who is saying that I'm not able to come on the video or all, okay? So please, contestant, keep this in mind. And a timeline has to be restricted to the 5 minutes only.
Hello, Miss Gargi. I am audible to Mr. Gargi.
Yes, sir, loud and clear. Sir, I'm just facing a bit of a glitch. I'm just joining my phone from my phone. Is it like permittable, sir?
Okay.
Okay, sir. I'm just sharing my screen here.
So mute yourself.
So good afternoon, everyone. And I'm sorry that I was late for my presentation because I faced some sort of a glitch. So I'll be giving my presentation from my phone. So am I allowed to share my screen?
Yes, please.
Thank you, ma'am.
I hope it is visible to you all.
It's not visible.
And am I audible? Just give me a minute, ma'am. How about now, ma'am? Can someone let me know, is my screen visible now?
It's not visible.
Can technical team check the permission? Do I have the permission to share?
Yeah, you are permitted here, but I don't think your slide is visible.
नहीं इससे आगे वाला जो है ना उसको लाइट कर दो पहले.
You have to broadcast. I think you are using the iPad, no? When you broadcast, then your camera,
Yes, sir.
then your camera will be closed. So just
All right, sir. So I think I should turn off my video because I think that would be a glitch from my side.
yes, you have to.
Okay.
Okay, sir. I'm so sorry, ma'am. I hope you got the situation that
Yeah. We can understand this.
Thank you, ma'am. Thank you so much, ma'am. Ma'am, is it visible now?
Yeah, but you have to close your other, other screen. You have to close. Too many screens are open. Yeah.
Dear participant, please focus only on your objectives and results.
All right, sir. I'll keep that in mind, sir. So good afternoon, everyone, and it's an honor to present my work in here that I have done in my previous years. So my topic of work is establishment of network construction imposition and finding the alkyl efficacy that could be targeted in a therapeutic way to deal with the dementia particularly. So introduction. So as we all know that dementia is affecting globally people with 55 millions of the population, and that could entitle it as a global concerning effect for the humans. So it have two types of prevailing. One, it is like sort of a thing. And that leads to a cognitive decline and a neural efficacy. So in another slide, we can see that there are like two brains. One normal, another one is dementia brain. We can clearly see by the observation that there is a sort of a shrinking. And why this shrinking happens? It's because of the three molecular hallmarks. One, beta plaque deposition and neurofibrillary tangles. And the third one is dysfunction of chlorine neurons in Alzheimer's diseases. So what is the pathophysiology of these all three? So we can see in the further slides that a neural membrane of a brain where is a APP produce amyloid that is equally important for the maintenance and proper functioning of the brain. But as like other cells, it need a restoration and sort of a refunctioning to regain its ability. So it need an updation. So there are like two enzymes called alpha and gamma secretase that breaks into a fragment, and this process is known as a non-amyloid pathways, amyloidogenic pathway. And second way, it is breakdown by amyloid beta plaque that breaks into a 36 to 43 amino acids monomer that is known as amyloid beta monomer. So this monomer actually blocks the axon terminal of the peripheral neurons and the dendrites of the postsynaptic neurons that leads to the hurdle and non-communication between the neurons and causes a degeneration. And second is tau protein binding. So there are microtubules which are important for the translocation and transmittance of the nutrients and and other secretions. So it is being binded and stabilized by the tau protein. But when when there is a disablation in tau protein, there's a hurdlance, and that will leads to synaptic hurdles that we can see as a cross in the picture. It is also known as neural junctions, and neural junction is disrupted by deposition of the tau protein. So this is like the pathophysiology of the Alzheimer. So in my studies, by using computational biotechnology and bioinformatics, I collected the data of Alzheimer and normal patients, and then I went for a protein interaction analysis. And before that, I find some of the differential genes in which are upregulated. That means they're highly expressive in Alzheimer's patient. And one of the gene that have been highly expressed was HSPA5. HSPA5 is actually a good gene because studies have seen that if we increase the expressivity of HSPA5, it can eventually slow down the neurodegeneration, as well as it can also release some ER stress. So these two, and ER stress is very important when it comes to any sort of neural dysfunctioning or any sort of brain hurdles, because that's where misfolding of the proteins happens. So this have been sound promising. So in my further studies, I was like, so how we can just increase the expressivity of HSPA5? It was by manipulations of the area where I have to only go for algal phytochemicals. So in algal phytochemicals, I have created a library of I hope it is visible, 12 total phytochemicals, in which I tested the binding affinity of these phytochemicals with HSPA5 secretion protein, that is also known as GNSCR proteins. So bromophycolide A, which have binding affinity of -12.7, have proven a promising when it comes to the binding affinity with that particular protein, and that leads to a hyperexpression of the HSPA5 that will further leads to a kind of a strategic approach by the expression of the Alzheimer's diseases and its regulation. So but it's not always about if we have the solution, we have to also focus on how economical and how available that particular solution is. So HSPA5 by testing a binding affinity with bromophycolide A, we have came to know across that there are already existing solutions, as we know that we cannot stop or reverse the neurodegenericity. We can only treat the symptoms. But this bromophycolide A have also seen that we can also add one more feature in a drug therapy, that is neuroprotection. So that's why it is very essential to go for further research in algal efficacy of phytochemicals and when it comes to Alzheimer's. But we have some restrictions. For example, economical variability and availability. Like every other drug, it is not marketally available. It is under the research still in itself. So that's make it more challenging, because the mining of this thing would be very hard, and making it publicly available would be a new challenge, because there have been already drugs that are showing some of it a better improvement when it comes to treating symptoms, but not the disease. But then again, need for this, why we had a need for this alternative, because it have a potential yeah, it have a promising potential to reduce the inflammation. Inflammation is also caused by dysfunctioning of neural mitochondrial membranes. And bromophytochemical studies have shown that it could be a promising approach by decreasing that. And for all these work, my methodology was typically, collecting data and then conducting a statistical analysis by using R Studio and R language. Specifically, if I mention the package called bioconductor and DE-seq analysis, and in that I have find upregulatory gene and
Sorry for
downregulatory.
interruption. I think
I think
you have
you
already
have
taken
already
too
taken
much
too
of
much of
time.
time.
So
Oh
So
yes,
please
sir.
please
brief
brief
within
brief
few
within few
seconds.
seconds.
All right, so thank you for letting me know. So this was my work. Technically, I'm winding up now. So from all this presentation, I can conclude the points. Yes, bromophycolite A have a promising and futuristic advancement because it also add one more feature, neuroprotection drugs in the future. So it could be a sort of an enhancement. And second thing, all these work were prevailing and like very ethical because bioinformatics and programming and biotechnology also reduce some sort of animal abuse and animal sacrifice. So that's how I wind up my presentation. And thank you so much for your time, so ma'am. I'm up for a question if there's any.
Any question from the audience?
Gargi, it was very nice presentation, though I know there was a constraint of timing, right? But your work is very nice, I think, right? So if the audience there have some questions or the query, you may raise these because we are already late by because you know her she was busy in due to the network glitch. So let us have some few questions because we have the marks for the question as well. Okay.
All right, ma'am.
Audience, any question?
I think, sir, there is like question and answer session also. So the participant has to be very alert because when they are putting the question to the presenter, it will be counted. Okay. So the question has to be raised from the audiences as well.
Yes. Yes.
Participant, especially the participant. Any of the contestants who are willing to ask the questions?
Madam, if no, then chairs and judges can take liberty to assist her by putting their questions.
Okay, ma'am. Thank you. Gargi?
Yes, sir. Good afternoon, sir.
Good afternoon. Which cell you have to treat this compound?
Sorry, sir. Can you repeat the question again?
Which cell line you are going to use this for this study?
So cell line. Okay. So for the I think your question is pretty much of a production specific, which cell line we could have been using. I'm not technically aware about it, which cell line we will be using, but there are like some animal modeling as well, where we can directly see the efficacies. And since not like a since already have been research conducted on bromophycolide A. So I think that could make it eligible to go for a like animal modeling testing in itself. So
You starting from the animal model, not from the vitro model first.
So vitro model, we see the efficacy of affinity binding. That is technically a ligation of a ligation between a ligand and a protein. So in my case, the HSPA5 secretion proteins and it have been ligated with bromophycolide A. So that was my virtually.
Thank you, guys.
Thank you.
Next participant.
Okay, sir. Thank you, sir.
Priyanka?
Are these participants absent or they are not in the presenting as their work? Ma'am, Dr. Prachi?
Madam, they have given their abstract and they have given their name also for the presentations.
So they are not present here, ma'am?
I think so. So you may continue to the next one.
Right.
Next, Dr. Jyoti Prakash?
No, she's in the DRC, so she's not going to present.
She's not.
Another again.
Brankuros.
Brankuros.
Hello. Hello.
Yes, Prankur, you are audible. You are audible.
Good afternoon, ma'am.
Good afternoon, Peter.
Good afternoon, all of you. My name is Prankur Avasthi.
Good afternoon. Go ahead, please.
Yes.
Prankur, go ahead. I'm audible?
I'm sharing screen now.
Okay.
Is it visible?
Yeah, it's visible.
Oh, sorry. So, my name is Prankur Avasthi. I am working under Dr. Sabha Hassan ma'am, and my today's topic is decoding DLBCL, key genes, and pathways in the spotlight. The diffuse large B cell lymphoma is characterized by the diffuse expansion of B cells with nuclei larger than 2 lymphocytes. It accounts for approximately 40% of all B cell lymphomas, making it the most common form of the non-Hodgkin's lymphoma. It is highly heterogeneous disease. Often exhibiting distinct genetic and molecular profiles. Around 60% of DLBCL cases responds to the RCHOP therapy. Many patients develop resistance to RCHOP or experience relapse, leading to poor outcomes. The exact cause of DLBCL remains unclear, but is associated with genetic aberrations like Epstein-Barr virus and other factors. It is crucial to establish a mechanism for identifying the main or core genes for better early identification and prognosis of DLBCL. So my objective is was that identification of critical genes which involved in DLBCL, analyze protein-protein interactions and functional enrichment to determine key molecular mechanisms, highlight potential therapeutic targets for overcoming drug resistance and improving treatment strategies. So this is my workflow of this study. So I have extracted the genes from this GeneNet and gene cards. We combined the genes and extracted the common genes, overlapping genes, which is 144 overlapping genes. Further, we see the functional enrichment analysis in which we perform the gene ontologies and GAC pathway analysis. And further, we see the protein-protein interaction analysis, in which further we see the what are the top 5 key genes in this. So this is the Venn diagram, which is made from the Venn 2.1. So 144 genes extracted, overlapping genes, and this is the CAC pathway analysis, in which we can see that the most of these round shapes are different round shapes, which is associated with the number of genes, and the cancer related pathways are their JAK state signaling pathway, Epstein-Barr virus infections, pathways in cancer, PI3K AKT signaling pathway, which is showing that these genes are involved in the cancer progression. And the biological processes, which is saying that B cell activation and lymphocytes differentiation, and most of these genes are related to the cancer, cellular in as well as in cellular component and the molecular functions. This is the protein-protein interaction analysis, in which I put that total combined score is 0.9, which is more highly which is the highest score, is telling that it is increase the confidence of the network. So in which there is a 143 nodes and 1409 edges. Edges is the connection between the predicting connections between the proteins, and 143 are the nodes, which are the genes. And this is the highly significant enrichment analysis. So further, I performed the k-means clustering, which is a unsupervised learning, and it made the nearest centroids data points, that is proteins, which proteins make the nearest connections. Further, I put these genes in the cytoscape plugin cytoba 0.1. I found that these are the top 5 genes which is based on the degree of centrality. And further, I saw that these genes are STAT3 related to JAK state, directly related to JAK state pathway, and PI3 AKT pathways, which is causing drug resistance and B cell development and cell proliferation and apoptosis. So I forgot to mention that the cluster 1 is also showing that these genes are present in the cluster 1, and for targeting this cluster cluster genes would be the beneficial for treating the DLBCL. So this is my conclusion, that these findings that suggested that these genes could be useful in that therapeutic targets in the DLBCL, and these two pathways, AKT, PI3KT, and JAK-STAT pathway, signaling pathway, may provide a new treatment strategies for overcoming drug resistance in DLBCL. So for future studies, should focus on clinical validation and drug screening to assess the therapeutic potential of these targets. Thank you.
Thank you, Prankar, for timely completing.
Hello.
Rahul sir, you are not audible. Please unmute. Okay, Prankur, one question from my side. What is the value of K in your project?
K means?
What is the value of K? For K means clustering, what value have you
this
taken?
is the unsupervised clustering. This is a random clustering.
No, that I understood. My question is, what is the value of K? Like when we do K means clustering, we decide the value of K. So what value have you taken?
Ma'am, it is a random kind of clustering. So
No.
This
I think you are not getting me.
Yes, ma'am.
Whenever we do K means clustering, first of all, we decide that the value of K will be 3, 5, 6, something.
Yes, ma'am, 3.
You have taken 3.
That was 3.
Why?
Yes.
Why 3?
Because I saw most of the papers in which they use the 3 value.
Okay.
Yes.
So you can try something else also. And what particular distance metric have you utilized for calculating the cluster?
Yeah.
Like in K means clustering, we take a distance metric.
Yes, ma'am.
So which metric have you used?
Ma'am, this I forgot, but
it's okay.
Yes, ma'am.
Okay, thank you.
Thank you, ma'am. Thank you.
Prankur, this data you have done some work into the wet lab?
Yes, sir. We are going to work on the patient sample to see the expression of the genes.
How?
With RT-PCR and Western blot further.
Western blot.
Yes.
So how can you say that your drugs is affected to this pathway?
Further, we can check the in vitro. In vitro, we can check the action of the drugs.
Which cell line you have to use for this experiment?
Sir, 2PK3.
Which?
2PK3.
2PK3. Is this specific for this receptor?
Yes, it is a DLBCL cell line.
There is a IP3 hydrolysis is going on?
Yes, sir.
This is the inositol triphosphate is going on?
Sir, I'm currently not using, but we are planning to do.
Okay.
Yes.
So is there any specific receptor is present on the surface of that cell line?
Sir, I don't know about this. Sorry.
Okay,
Yes.
so before you have to perform your experiment, at
Yes,
least
sir.
you should go that which cell line is suitable for this experiment. Otherwise, this is the common pathway which is available into all the cancer cell line.
Yes.
Okay.
Yes, sir.
So thank you for my side. Okay,
Thank you, sir.
Prankur?
Thank you, sir.
Yes.
Our next participant, was Nakvi or Prekshigar.
Yes, sir. I hope I'm audible. Hello.
Please be precise.
Yes, sir. Sure. I hope I'm audible.
Yes, Vyas, you are audible.
Okay, ma'am. And I hope my presentation is also visible.
Yeah, please put it in full screen mode.
Sure, ma'am. Okay. Good afternoon to one and all present here. Today, I, Y.S. Naqvi, will be giving a presentation on the topic Metagenomic study reveal Akkermansia mucophilia to be negatively regulated in gut of patients suffering with psoriasis.
Sorry to interrupt. Why, if possible, please switch on your camera.
Okay, so just a second, I'll try to. So actually, the thing is that I have no light in my area, and if I turn on the camera, my laptop's battery will die out, and I cannot charge it.
Nakvi, you were aware na that you are having presentation today at this time?
Ma'am, actually, there's no light in my area. I was aware, but there's no
You
light.
don't
Yeah.
have the backup?
Ma'am, actually, it's on battery saver mode. I'll just turn
Okay.
on. Yeah.
So
Okay then.
voice connect
Go
with
ahead.
your mobile also so that
Go
you can
ahead.
switch on the
Okay, go
mobile's
ahead.
camera.
Okay, ma'am. Okay. So, ma'am, shall I start or should I just join with my phone as well?
Okay, YS, go ahead,
Yes, sir.
please.
Okay,
At
okay,
the
so sorry.
end of that, you can.
Okay, sorry for the delay. So, psoriasis is a chronic skin inflammatory skin disease characterized by uncontrolled keratinocyte growth and altered differentiation caused by chronic inflammation. Psoriasis affects a total of 125 million people globally, making up to 2 to 3% of the total population. Gut microbiota refers to all the microorganisms that inhabit the gastrointestinal tract and includes bacteria, archaea, and eukarya. It shares a somewhat mutualistic relationship with its host and plays a crucial role in maintaining gut epithelial barrier integrity, defending against harmful pathogens, and regulating immune responses. The relationship between gut microbiota and immune system helps in understanding the pathogenesis of many chronic diseases, including psoriasis. Gut microbiome dysbiosis is a condition wherein the microbial composition becomes altered, which eventually results in the activation of the immune response via the interleukin 23 and interleukin 17 pathway, promoting skin inflammation. This is the methodology that I followed in my research, and this is referred to as the QIM2 pipeline, which is a microbiome analysis package utilizing the 16S rRNA gene sequencing. So the first step was to collect the raw sequences, which were obtained from stool sample of 63 patients and 54 control from gene expression omnibus. Then, following the raw sequence collection, pre-processing was done, which involves demultiplexing and denoising. In demultiplexing, we sort the sequences back to their original samples based on the unique barcodes provided to them. And in denoising, cleaning and removal of errors is done to get accurate DNA. It helps in filtering out low quality reads so as to ensure that the most reliable information is retained for the analysis. Diversity in terms of microbiome is of two types, alpha diversity and beta diversity. For beta diversity, a box plot was created and it was interpreted from this box plot that in the control group, the diversity is lower as compared to the diversity in the patient group. This finding was further supported by the Kruskal-Wallis test, which yielded a p-value of 0.002834, which means that there exists significant difference in diversity of both patient and control groups. Then, for the beta diversity, we looked up at the unweighted UNIFAC PCOA plot for visualizing differences between the control and patient samples based on the phylogenetic distances. In this plot, we can see the red dots and blue dots. The red dot denote the control, while blue dot denote the patients. Blue dots in here are tightly clustered, meaning that they have similarities in their microbiome, while the red dots are more spaced out, meaning that variability exists within its microbiome. To further support our findings from the beta diversity, Adonis test was done, which yielded a p-value of 0.001, indicating that the microbial composition is statistically significant and meaningfully different. This is a bar plot wherein on the y-axis we have the frequency and on the x-axis we have the control and patient groups. We look up at it to check the composition and diversity of bacterial communities within the patient and control groups. However, to look up at the specific microbes that are being upregulated or downregulated during dysbiosis, a volcano plot was used, where on the y-axis we have the CLR and on the x-axis we have CLR and on the y-axis we have W statistics. In this plot, one dot is located to the far left, which I have highlighted with a yellow circle, which is the bacterium Akkermansia muciniphila that is significantly more abundant in control group compared to the patient group. This means that this bacteria is getting downregulated in people suffering with psoriasis. The analysis of gut microbiome identified that the gram-negative anaerobic bacterium Akkermansia muciniphila was reduced in individuals suffering with psoriasis. Akkermansia muciniphila comprises of anti-inflammatory properties via the production of short-chain fatty acids and reducing pro-inflammatory cytokine levels. Akkermansia muciniphila promotes gut barrier integrity and regulates the adaptive and innate immune responses. Hence, when the level of Akkermansia muciniphila are reduced during gut microbiome dysbiosis, lower levels of SCFAs are produced, which disrupt the intestinal barrier, allowing the translocation of bacteria, that is the leaky gut, and along with an heightened immune response, ultimately resulting in psoriasis. These are all the references, and this is it from my side. Thank you everyone for your kind and patient listening. Thank you. And I'll just turn on my camera, just a second. I hope I'm visible. Sorry, actually there's no light, so just turn. Yeah, sorry, there's no light.
Dr. Meenakshi? I think she is not here. Voice?
Yes, sir.
This is the gram-negative bacteria, right?
Yes, sir.
So you are saying that this is a good for, as like the probiotic type.
So basically, it is found in the gastrointestinal tract of humans, and from my studies, it was concluded that it is an SCFA producing bacteria, and it basically controls the pro-inflammatory cytokine levels and basically it controls inflammation. So it is getting reduced in patients with psoriasis. So that is why we observe psoriasis in people.
Do you think so? This is the right? Because this is gram-negative bacteria, they are producing some LPS is there.
Mm-hmm.
The
Yes,
LPS is there, then the inflammation is going on and the psoriasis is again, right?
Yes, sir.
So how can you justify your study if it is reaching into the gut and they can do something better condition?
So basically, this is already present in the gut, and my findings have identified that people with psoriasis have reduced levels of this specific bacteria. So
what is the mechanism behind that? If I ask you.
So the mechanism, if I were to talk about it, I mean, I didn't exactly look up at it, but the thing is that what I have done in my study is I have compared the samples from people suffering with psoriasis and I got this result. Yeah, but I need to do further study on this to understand the exact mechanism. But this is like the starting point. It is forming a link that
okay. That's all from my side.
Okay, so
that's all from my side. I think
okay, which particular tool have you utilized for your study?
Ma'am, basically the tools that I used were Quant2View and Galaxy Europe server. And basically both of these were servers wherein and NCBI as well. So basically data retrieval was done from NCBI and GEO. Like data sets were retrieved from them, metagenomic data sets. And then I uploaded them on the Galaxy Europe server to like pre-process them. And then after pre-processing, I got those files, the QZA and QZV files.
My first question is, how did you remove the noise? Like in pre-processing, what have you done to remove the noise
Ma'am, to remove the noise, basically,
or the missing values in the pre-processing? What have you done for that?
ma'am, I cannot actually recall at this moment.
I
But
think you are not
yeah,
getting my question.
yeah,
How did you handle the missing values?
yeah. Normalization, you mean how I've done the
Yes,
normalization
yes,
process?
yes.
Yeah. But ma'am, I cannot recall that at this particular.
Okay, it's
Nakvi?
okay.
Yes, ma'am. Yes, Shalini, ma'am.
Yeah, and even to the judges or the biochemist people, just a query here that instead of going for the skin sample, why you went for the stool sample for the analysis right now? Because psoriasis, to the
It's a
best of
skin
my
disease.
yeah. So,
Yeah.
what motivated you to go for the please, sir, go for the collection of the stools and then doing the study that get gut microflora? What
Yeah.
is the correlation between that? How you have connected these two fields?
Ma'am, because like if I were to talk about it in a very honest manner, ma'am, I was very interested in studying metagenomics and performing microbial dysbiosis. So like when I looked up at the data sets, like usually I was getting data sets from skin only because psoriasis is a skin disease. So obviously you'll
Right.
get samples from skin only. But it was I had this idea that I want to connect the gut and psoriasis. कि अगर आपके what you're consuming because of that imbalance हो रहा है and because of that imbalance it is psoriasis getting caused basically inflammation हो रहा है तो मतलब मैम in my findings also
This is
I
the so you see, this is the second, you can say that this is the secondary infection, not the primary infection. The primary infection is on the skin. That is the cirrhosis is the condition where the infection has occurred through the skin. Some type of the there is a problem with that, and then you went for the I
study.
still cannot understand if the other expert uh, who are from the biotechnology field. So I am not able to understand, is this content like no?
What's the ground level approach
Yeah.
that why I've decided this? Yeah.
Yeah. Okay. Anyways, good one, right?
Good one.
Yeah.
Good one. Okay.
Thank you, ma'am.
Next in line is Mr. Isan Rastogi.
Good afternoon, everyone. Am I audible? Hello, sir. Good afternoon, sir.
Ishan, share your screen. Your presentation is not visible.
Yes, ma'am. Just check, ma'am. There's some issue with this one. Just a moment, ma'am. Yes, ma'am. I'm audible, ma'am. Okay, ma'am. So, I hope it is visible. Am I audible, sir?
Yes, Ishan, you are audible.
Thank you so much. Yeah. So, abstracts what I'll be explaining today. My topic is proteomic analysis of chronic obstructive pulmonary disorder. Sorry. So, first of all, let's start with what is COPD. Chronic obstructive pulmonary disease is a lung disease that causes breathing problems due to long-term inflammation. It involves proteins like elastase, alpha-1 antitrypsin, and inflammatory cytokines like IL-6, TNF-alpha, which damages the lungs. So we analyze that the lung tissues, fluids, and blood samples to find the biomarkers that are the protein linked to COPDs. A computer model of a RAT protein was created from the Swiss modeler to study the disease. I used the Ramachandran plot so that we could check the accuracy of the protein model using the safe server. I found out that the few proteins are linked to oxidative stress, which are the damaged cells, and immune responses. We also find out the mitochondrial dysfunction, which is the energy problems in cells, and metabolic changes play a role in COPD. Biomarkers help predict diseases progression and identify the potential treatments. And faced a lot of challenges during the research, which was differences in patient samples, making the results vary. Secondly, technical limitations in studying the proteins. And the third was the data integration, which is combining different research methods. My further research on this was that scientists are trying to combine the proteomics with genomics, which is the protein plus gene studies for better understanding for the disease, which could lead to early detection, personalized treatments, and improved outcomes for the COPD patients. In short, the COPD is a serious lung disease, and the scientists are still studying the proteins to understand how it worsens it. They use advanced tools and computer models to analyze lung samples. Although there are challenges, future research which could lead to better treatment and early diagnosis. Thank you.
Judges, you may ask your questions.
Hey, Ishan, what are you doing right now? Are you a student or what?
I am a student, ma'am,
Of which course?
of BSc Biotechnology.
So why have you not made a proper presentation? Why are you presenting in Word file?
Actually, I wrote the abstract itself, so I just presented the abstract.
Okay, okay. So at least some data, some results should be there in the form of, so we can understand, like only theory, it was difficult to understand what you want to. Can you summarize your work in 1 or 2 lines?
Okay, ma'am. So, like after summarizing it, I was okay, ma'am. In short, I would just say one thing that the chronic obstructive pulmonary disease, it is a lung disease, and of course, the scientists are like still trying to find the proteins, and they're trying to find how it worsens it, how it worsens the disease in the body more.
It's fine.
So
What's
they
your
are.
contribution? What's your contribution?
So, ma'am, actually, I found out the results using the Swiss model, and I got the template, and through that I used the safe server, and I found out few results in Ramachandran plot as well. So yeah, I did that.
Okay, thank you, Ishan.
Okay, thank you.
I'm not able to hear, sir.
Our next in line is Mr. A. Ramkrishna or Faizan. The topic is Squalene C produced by Bacillus species.
Yes, sir.
Who will present this work?
Yes, sir, Ramakrishna.
Okay, go ahead.
Thank you, sir.
Rahul sir, this name is not in the judgment sheet. Rahul sir?
It is there.
Ma'am,
It is
ma'am, maybe
last,
some of the
in last three. In last three. His name is in last three.
Yes, ma'am. Ma'am, shall it start?
Yes, please. You have only 5 minutes.
Thank you, ma'am. Respected dignitaries, estimated panel members, honorable judges, dear colleagues and friends, a very good afternoon to all. I am Arsvili Ramakrishna. It is a pleasure to be here today to discuss my research on Squalene C produced by Bacillus. Effectively inhibit sucrose transporter SRT1 essential for Stelgomyces virulence. It has a great significance in global agriculture plant pathogenic fungi and their impact on crucial crops like rice and maize. These are the two of the most essential crops worldwide, feeding billions of people. However, their productivity is constantly threatened by fungal pathogens that not only reduce yield but also compromise food safety and quality. Among these, Stalagmites stand out as a major concern due to their unique
Sorry to interrupt.
Hello. Stalagmites, it it
Your slides are not changing.
Sir? Sir, अभी Yes, sir. Sorry, sir. Initially,
Okay, go ahead.
Initially considered a minor disease, now emerged as a global threat, particularly in regions like India and Mid East. This disease leads to the formation of false mud balls on rice grains. Now coming to the objectives. At first, homology or AB initio modeling of SRT1 protein structure and visual screening. Third, molecular docking, and fourth, molecular dynamic stimulation. Methodology. Structure modeling. Upon reviewing the literature, we found that SRT1 gene is critical to virulence of Stelgomyces. There is no proper structure was available. Hence, we went for the AB initio modeling of protein sequence to design structure-specific inhibitor. We use FIRE 2.2 tool to predict its structure. Coming to the virtual screening, initially 36,454 natural compounds which screened for the natural product atlas database. After that, 485 compounds screened has come to the target naturally compounds produced by the bacillus. From that, using the Limpinski rule of 5, screening down into 309. Out of that, 198 compounds finally screened successfully. After that, 198 after that, using camera 1.18 version, we went for the molecular docking and finalized the inhibitor based on the binding affinity. Coming to the results, now this is the three-dimensional structure of protein. When we validate it through Ramachandran plot, we just see that 89.2% core region and 10% where is the allotted region, hence we use for further. Molecular docking. The top template includes human glucose transporter is with 84% coverage, 100% confidence. Pro-assay analysis show that protein folding energy of the model is good agreement favoring the validation of the model generated with the score of minus 4.11. And this is the table shows the top 5 inhibitor of SRT1. And this is the structure of SRT1 gene interacted with the Squalene C ligand. Molecular docking performed by all the 198 ligands with the SRT1. From the docking, determined 5 ligands to have the highest bonding affinity to the SRT1, which varied to range of minus 10.5 kilocalories per mole to minus 10.9 kilocalories per mole. We select the ligand spirulin C based on the high binding affinity minus 10.9 kilocalories per mole. Now that we successfully identify the suitable protein, we did it further validating using MDS, molecular dynamic stimulation, over 100 NS. Now these are the RMSD and RMSF plot of study which show that inhibit bonds protein at active site very strongly. Now we successfully conclude that SRT1 gene inhibited by the spirulin C without affecting the plant sucrose transporter, and this approach is eco-friendly. Discussion. If we use
Okay. Thank you, Ramakrishna. You have already utilized 10 minutes. So session is open for question and answers.
Ramachandran? Ramakrishnan?
Yes, ma'am.
That's okay. So, do you know what this is commonly known as the disease of the maize?
Yes, ma'am. False smut disease.
False smut disease.
Yes.
So, does this happen in other crop also other than maize?
Maize and rice, ma'am. Actually, majorly maize and rice.
Maize and rice. So, what had forced you to take this study? First one. What was the motive of taking this study? Have you done some of the, references? Have you collected some references for this?
Yes, ma'am. Actually, my project includes the development of bio-based antifungal treatment for agriculture.
I know, but why did you took this specific fungus or you specific disease?
It causing most.
Yes.
Most of affected in maize.
It is most virulent one, and it is heavily destroying our rice crop, right? Okay, so you are recommending this as the futuristic scope for the bioagent, right?
Yes, ma'am.
Okay, can you please highlight your findings? Highlight your findings, specific one.
Spirulin C, ma'am. As a ligand to inhibit the SRT1.
Yes. Okay.
Yes.
So you're not very clear with your findings, right? Is this true? Or you don't want to disclose it here?
No, ma'am, actually.
Okay.
Any other question, judges? Audience?
Sir, I have one question. I'm Kiran.
Okay, go ahead, Kiran.
I just wanted to understand that molecular simulation. So after your docking, like for molecular simulation, how you did it and how much time actually it took for you to conclude your result after simulation?
Ramakrishna, you can answer.
Yes, sir. Yes, sir. Ma'am, could you please repeat the question?
My question is after docking.
Yes, ma'am.
The next step was simulation.
Yes, ma'am. Simulation.
So simulation after docking, like how much time it took for you to do that the simulation part completely? How you got results? Was it like a very time-taking process or lengthy process, or like how many
I took the help
days or months you took?
I took the help of my second author, ma'am. Like from
okay. Understood, sir. Thank you.
Thank you.
Thank you, Ramakrishna. So now we have Dr. Monica Kamoj, ma'am, with us. So her topic is application of molecular modeling. Ma'am. Are you there? Dr. Monica? Okay, so we can begin with another participant. Mr. Abhishek Nandi. Are you there?
Hello.
Okay, please share your PPT and switch on your camera. Turn on your camera, please, and start your presentation.
Sir, I'm visible.
Yes, go ahead.
Sir, is my presentation visible? Okay.
Yes.
Okay. Is my presentation on full screen mode? No? Thank you, sir.
Yes, go ahead. All the best.
Good afternoon, everyone. I am
Abhishek, you are not audible.
Hello. Hello.
Please go ahead.
Okay, go ahead.
Good afternoon, everyone. I am Abhishek Nandi, and I am delighted to be presenting my research on a fascinating and crucial topic, an in silico approach for predicting antimicrobial properties of fast lytic protein. We are living in an era where antimicrobial resistance is one of the greatest challenges in healthcare. Acinetobacter baumannii, a highly drug-resistant pathogen, is a major cause of hospital-acquired infection, mainly causing lungs and wound infection. But bacteriophages are viruses that specifically attack bacteria and offer a promising alternative to antibiotic treatment. Instead of relying solely on antibiotics to combat antimicrobial resistance, we can explore fast lytic proteins as a powerful antimicrobial agent which has antibiofilm properties with high cellular penetration and has less toxic effect on humans. Here is a list of different types of lysins, and the most common ones are endolysins, lysozymes, and carboxypeptidase. In this presentation, I will talk about how we use in silico method to predict the antimicrobial potential of these fast lytic proteins, proving a new pathway for drug discovery. In this methodology, first the sequence of the fast derived proteins were retrieved from FALPI database. Then identification of antimicrobial peptide was done using AMP Discoverer tool. Then the antibiofilm activity was done by DPABB online tool. Next to that, cell penetration property of the peptide was predicted using cell PPD tool. And at the end, cell toxicity of the peptide was done using ToxSpread tool. Along with that, we have also predicted the structure of this fast lytic proteins using the Swiss model server, and the structure validation was done using SAFE 6.0 server. This is the result that I have got from the antimicrobial peptide prediction and their antimicrobial properties. Out of that, we have seen that endolysin and chitinase and the carboxypeptidase shows good result with cell penetration properties, and they are non-toxic also with a good SVM score, and lysozyme also show active antibiofilm property with less toxicity. This is the structure prediction result of all the 5 peptide that has that has antibiofilm properties, and then the Ramachandran score plot percent with the favor percentage score also. So in the interpretation, you can say that carboxypeptidase, endolysin, and this lysozyme with endopeptidase show the anti-biofilm properties. Endolysin, chitinase, and carboxypeptidase have shown good cell penetration property, and almost all the peptides are observed to have non-toxicity towards human. Endopeptidase and lysozyme show maximum percentage with the template model when predicted in the Swiss model server. And in the conclusion, I must say that fast lytic proteins offers a promising targeted alternative to antibiotics with strong antimicrobial and antibiofilm properties. This research is crucial step towards developing most alternative to antibiotic resistant that is coming up in the next few years, and to combat the drug resistance that Acinetobacter baumannii is going to show. Thank you. This is the reference.
Abhishek for presenting within given time limit. Now session is open for question and answer. Honorable judges, you may ask your questions.
Okay, Abhishek.
Yes, ma'am.
Which in silico technique have you applied?
Ma'am, I have used prediction tools like the prediction tools that I have used are AMP Discoverer. Like first, I have retrieved the amino acid sequence for this proteins, and then I have put the sequence in the AMP Discoverer tool. This uses SVM matrix to predict the particular sequence for the like which part of the peptide is shows antimicrobial property. This
Okay, have you validated your work also, or it is only till now it's only in silico method?
Ma'am, in wet lab you are telling?
Yeah.
Most of my work is in wet lab only. So I have not done it with proteins, but I have done it with bacteriophage. In bacteriophage, I have done it with bacteriophage.
Thank you.
Thank you, ma'am.
Hello, Abhishek.
Yes, sir.
You are using these peptide as it is present into the bacteriophage.
The bacteriophage, sir, releases these peptides at the end of their lytic life cycle. So they are not produced inside the bacteriophage, they releases inside the bacteria. When their lytic life cycle gets over, they releases to kill the bacteria and get released.
Kill the bacteria and the bacteria will be released this peptide. You are saying that?
Yes, it is produced inside the bacteria, but we will isolate, like from the genetic material, we can isolate this and produce this peptide in lab also, in vitro also.
In vitro also. And they are affected to the other bacteria.
Yes, to this particular bacteria, like a bacteriophage produce the endolysin, and this will affect only that bacteria only, not to other bacteria. Like Acinetobacter phages will only affect Acinetobacter only.
Okay. Thank you.
Thank you, Abhishek.
Thank you, sir.
Now we have Dr. Monica Kamboj, ma'am. Are you there, ma'am? Abhishek, please unshare your screen. Thank you.
Monica, ma'am,
are you there? No, I think she is not there. Now,
next presentation is
of Kiran Kushwaha.
Her
Our
topic is identification of priority genes against ADHD by using network biology. Miss Kiran, please share your PPT.
Good afternoon, one and all present here. Thank you so much for the given opportunity. My today's presentation topic is identification of priority gene against ADHD by using network biology. I am a PhD scholar in SRMU Lucknow. ADHD we have all seen around, and they are not so noticeable, but it is a neural disorder. And globally, 7.2% of children and 3.4% of adults suffering from this order. So I use system biology approach. I'll quickly go to that slide. And in that, I collected data set in form of gene from genomics, proteomics, and transcriptomics. So these data set I collected through Malakart and GeneCart. This is my methodology. So after collecting these genes, I used software Cytoscape, and by using its string plugin, I tried gene network. So after gene interaction, I use CytoHub plugin and some other 10 to 12 other parameter. I found some 10 highly expressed gene. These 10 highly expressed gene, I again use I'm sorry. I tried checking the maximum expression. So on the basis of degree, I found a priority gene TP53. This is my string network. And these are the parameters which I have used, and I found 10 maximum expressed gene. Using this degree table, I found degree for TP53 was 110. So this gene TP53 is the priority gene here, and this can be a biomarker. This can be a therapeutic ligand also. So by identifying this priority gene, the further research are open. This can be tested in wet lab through PCR, and we can validate it, and we can further use it for drug development. So this is my small objective, finding a priority gene for ADHD. Thank you so much. Is there any question? I'm open for it.
Thank you so much. Now session is open for question and answer. Respected judges, you may ask your question first.
No question from my side.
It's a very
Audience, you may
small,
ask.
simple work. It's a beginning of this work.
No, it was good. It's good.
Audience, is there any question? Participants?
Miss Kiran.
Yes, ma'am.
I just wanted to know about because you say that this is not very common. ADMD is not very common, right? Only 2% people are affected by this, such kind of the neuro disorder. So could you please tell me the visible symptoms, how you are going to recognize these people?
Ma'am, actually, I'm an educator. So every year, 1 or 2 students I can see with ADHD, mild or hyper.
And what age group you come across?
Ma'am, if we see that identification, we can easily identify in like grade 1 and 2, but they go for the clinical.
And what is the criteria of grading? How we are going?
Ma'am, they are very hyperactive. They are very hyperactive. One. Second thing is they don't think before they do. They can do anything. Like very latest case, I will tell you.
If it is visible in 3-year child or a 10-year child, I just wanted to know this trend, because 3-year child, they are very hyperactive and they do not think before acting. So we cannot say that these people are affected by ADMD, right?
ADHD, yes.
Okay,
Ma'am,
so
the identification can start from grade 1 and 2, but we summarize it or we send for clinical assessment on around grade 4 and 5. When they are little mature, they can like handle the questionnaire session. There is a clinical assessment for them, and
say like 5 years and 6 years, basically.
Basically, ma'am, latestly I have identified one case in my class itself, and that child hyperactively, he burst one cracker in classroom, and it
Oh
was
my
so quick. Then and he's
I
very
just want to know the
intelligent.
age group, because this is, these kind of the these days, na, because parents, they do not give much time to their childrens. So the doctor
Yeah.
are saying that these can be the some of the cause, like for such notorious. First off,
Yes.
we mix them with a notorious child, right? And
Yes,
secondly, this
yes,
could
yes,
be a
yes.
mental disorder. So
When
that's
we
why
see
I was.
consequences. Yes, ma'am. I'm sorry to interrupt you. Please continue.
Please, go ahead.
So, ma'am, in such cases, we need parent consent, and then after they go for the clinical assessment. In that report, we can notice it, like whether it is a mild ADHD or it is a like severe ADHD, and then after they go for the treatment. But the treatment is very long.
Right.
So my interest in this is it's just one objective. Slowly I will come for the result where we can find some quick and affordable, or what we can say is there will be a lot of kind of side effects, because
This
these are
fall in
the
the biotechnology, or it falls in the psychological?
ma'am, it's a mixed, but like getting the result, then we have to go for the clinical assessment. This all is a medical requirement. As for my understanding and for my work is, I'm looking for some better treatment for this.
Okay.
So something if I can contribute.
So
So it
you
can
are
be.
going to identify gene as well, that which of gene is responsible for such kind of the mental or the health status.
Yes.
I should not
Yes,
use the
ma'am.
mental status for the such kind of. Okay. So
Yes.
neurogenes is affecting this.
Yes,
You will
ma'am.
identify that one, and then it can be cured or not cured. So
Then there will be some further research, like whether we can control it or how we can plan better drugs, something
okay,
like that.
very nice.
Thank you
Okay,
so much, ma'am.
thank you so much.
So, thank you so much, participants, for your very nice and precise presentation. So, now I invite Dr. Monica Kamboj, ma'am. Are you there, ma'am? So, this is last call from our side. Okay, I think she is not there. So, this in this session 4, total 7 presentations given by our participants. So now I conclude this session 4 of International Symposium cum workshop on futuristic bioinformatics, shaping young minds. So I thank Professor Dr. Shalini Visan ma'am, Dr. Meenakshi Srivastava ma'am for their deliberations. I also thank Dr. Vineet Awasthi sir, who is chairperson of this session, and rapporteurs Dr. Garima Awasthi ma'am and Dr. Paras Porwal. Thank you so much. Dr. Vineet sir, would you like to say something?
Thank you.
Okay, thank you. Thank you, Dr. Meenakshi. Thank you,
sir.
Dr. Shalini ma'am.
Okay, I will also like to appreciate the effort of all the organizers of the biotech division, and I will especially thanks organizer and Dr. Prachi Srivastava for giving us the chance to be here with you all people and to have such a good presentation from our participant. Thank you all.
Thank you. Thank you, everyone. So now I have to close this session and now move forward to the next session for that. Okay. Dr. Prachi?
Thank you, sir. Thank you so much, sir.
So
yes, we
hand
are.
over to you.
Thank you so. Thank you so much, sir, for gracing the podium and sparing the time. I'm really thankful to you, Rahul sir, Shalini ma'am, and Dr. Meenakshi. All of you supported so well, and the very knowledgeable and very fruitful questions. So thank you for all of you for engaging the session and chairing and reporters. I'm so much thankful to all of you. Thank you.
Thank you, ma'am. Once again. Now I have to leave this.
Thank you to all the presenters for their insightful research and discussions. Now we move forward to one of the most awaited moments of the event, the validatory session, where we will reflect on the key takeaways from today's event. We would like to welcome Dr. J.K. Srivastava, sir, Director of Amity Institute of Biotechnology.
Thank you, Janvi. Am I audible or not?
Yes, sir.
Hello.
Sir, you are audible, sir.
Hello.
Yes, sir. Yes, sir.
Okay, I'm audible. Okay, fine. So, by that way, we are coming to the end of this 2 days conference or symposia. I learned that a lot of oral presentations has been done by the students and the participants and the research scholars also. So that is very encouraging that the students have taken very active part in oral presentations, seminar. And also the very interesting part is that whole event is organized, conducted by the students, by the cooperation and by the involvement of the students. So that is a trend of MIT. For that, I congratulate the organizers also. And finally, I think that the results are likely to be announced. So I have the results with me for yesterday's oral presentation. For today's oral presentation, I am just, let me check. No, it is, I have the only yesterday's presentations result. So Dr. Prachi, today's results presentations result are awaited.
But sir, it is just we are calculating under calculation because just now I have received the judgment sheets from judges. So it is 100%, just in 2 minutes.
So anyway, I find that the participants are from the Tamil Nadu also, Tamil Nadu Agricultural University and CSI or CDRI, and also our own students and many other students from the many other organizations also. So that's a very means encouraging thing that the participants from all over the country have taken the part in this symposium, in this workshop. And so should I announce the first day result? Because both their results are 20th result and 21st result are separately announced. So should I go for the 20th?
Yes, sir.
Huh? Okay.
Yes, sir, you can proceed for the first day result.
So, okay. So for the first day, first, let me congratulate everyone, all the participants of oral presentation, because this is a formality that we decide the first, second, third. Otherwise, your participation itself is a highly encouraging thing. So let me start from the lower ladder. We are having at third position, Miss Shreya Singh from BSc Biotechnology, 6th semester, MIT Institute of Biotechnology. Ms. Shreya, congratulations.
Thank you, sir.
Yeah. Then, if we move ahead, we have another scholar from Tamil Nadu, that is Mridula. She is also sharing the third rank from the Horticulture College and Research Institute, Tamil Nadu Agricultural University. Congratulations, Mridula. And next is the second position or the first runner-up, that is Anugya Singh, a research scholar from CSIR. I think she is from CDRI or which?
IITR. She's from IITR.
Okay. The CSIR, IITR, Anugya Srivastava. She's on second rank. So congratulations, Anugya.
Thank you, sir. Thank you.
Okay, right. And first position holder, our first ranker is Miss Vaishnavi from BSc Biotechnology 6th semester, MIT Institute of Biotechnology. So Vaishnavi, congratulations. You did a nice job, and rest all other participants, you also deserve congratulations for taking active participation for this program, this conference, this symposium. So this is the first day's result. I think we have to wait for a few more minutes for the second day and final day results. So in the meantime, I'd like to, if the Vaishnavi is there, Vaishnavi, are you there? No. Anugya, I think Anugya Shrivastava is there.
Yes, sir.
Okay, Anugya, so can you tell us about yourself in a very brief manner?
Yeah, sir. Actually, myself, Dr. Anugya Srivastava, I'm just completed my research from CSIR IITR.
Okay.
And I'm very much thankful to the organizer, especially Dr. Prachi Srivastava, who gave me an opportunity just to present my work in such a great platform. Thank you so much, sir.
So you are working in IITR as a right now, because you are already
As a
completed your
researcher
PhD.
right now, sir. As a researcher right now.
As a researcher.
Yeah.
So and with whom?
With Dr. Vinay Kumar Khanna.
Oh, Vinay Khanna. Okay, fine. Good. And what is your interest area?
Sir, neurotoxicity, neuroscience.
Of course. The guide is from the neurotoxicology, so you must be from the neurotoxicology. Okay,
Yes.
congratulations.
Thank
And
you, sir.
I wish you very good luck for future
Thank you.
endeavors. Right.
Thank you so much, sir.
Then we have Mridula. Mridula, are you there? Mridula, are you there? Yeah. You are on the screen, right? Because I don't see the name. But I hope that you are Mridula S. from Horticulture College of and Research Institute, Tamil Nadu. No, she's not there. Then Shreya is there.
Yes, sir.
Yes, Shreya. You are from the host institution, from our own department. So just tell us about yourself in a very brief manner.
Yes, sir. Thank you so much, sir. I am Shreya Singh from BSc Honors Biotechnology 6th semester.
Your camera is not operating. Just open your camera.
So
Yeah.
actually,
Oh, you got
I
injury.
have a medical condition, yes, sir. So that's
Oh,
why.
very well.
So actually, I am really grateful to Prachi ma'am, who helped a lot along with the session also. And this was my first time of presenting some paper or something. And I'm really happy that I got a position. And this helps me and motivates me more and more. And I'll look forward for more paper presentation and another works.
You are in BSc 6th semester, final semester you are.
Yes, sir.
So
Yes,
after
sir.
that, what's your plan?
So I'm going to do MSc, and for that I'm preparing for the entrance exams such as GAT-B and CUET PG.
Okay. You got a very serious injury on your just below the eyes.
Yes, yes, sir.
Yes. Anyway, good luck.
Thank you so much,
Yes.
sir.
Right. So,
Sir, I'm just forwarding you results. Results are with me. I'm just forwarding you.
okay. Yes. Madam, you are forwarding on mail or WhatsApp? Raji ma'am?
Sir, just forwarding.
No, through mail or on a WhatsApp?
Sir, on WhatsApp.
Okay, that's why I'm just watching in WhatsApp. Okay. So I got the results for second day. Should I announce the results for second day? Means today?
Yes, sir. Yes. Sir.
Okay. At third position means second runner-up. There are two participants. Arsavilli Rama Krishna, very long name, and Abhishek Nandi, Mr. Abhishek Nandi. So congratulations.
Thank you, sir.
Ramakrishna and Abhishek.
Thank you, sir. Thank
Abhishek,
you.
are you there?
Yes, sir.
I can understand your voice. Any change? There is a little bit correction on the first day result. I'm sorry. But there are some good news also. With this correction, there is good news. Ms. Vaishnavi and Ms. Anugya Srivastava, they hold the first position. They are holding the first position. Mridula is, instead of third, she is holding the second rank. And Shreya is, as usual, she is on the third, means second runner-up. But this result is also shared by Sri Nivasu Patakula. So congratulations to all the participants, yesterday's participants. And now we are having today's result. So third, I have just mentioned that third rank is shared by Arsavalli, Ramakrishna, and Abhishek Nandi. Congratulations, both of you. Second position is by Kiran Kushwaha. Kiran, are you there?
Thank you so much, sir. I'm here. Yes, sir.
Yes. So, Kiran Kushwaha, you are there, right? And first is Vais Nakvi.
Second prize.
Vais, congratulations. Are you there? Vais, are you there? There? He's not there. Anyway, so we can have some feedback from the Kiran Kushwaha, who is on the screen. Kiran, please turn on your camera and just tell us about
Thank you so
yourself,
much, sir.
your feedback.
Sir, it was very, very motivating, and I can't express my feeling. I'm working in as an educator in Bangalore and pursuing my PhD from SRMU Lucknow.
You are
I'm
working
working on it
you are working in Bangalore pursuing
as
PhD from
educator. Yes, sir. PhD Lucknow. Yes, sir.
what is that equation? I don't understand. Means
Uh,
I think you have appeared in your PhD entrance examination also here
uh,
in MIT.
no, not exactly, sir.
No? Okay.
No.
Anyway. So you are currently at SRMU Lucknow or in...
Sir, you are
No, no,
very
sir,
right. Sir, she appeared, whatever I remember, she appeared during the GRF interview that what we have conducted.
yes,
Yes,
yes, yes,
yes,
yes.
sir,
I
yes,
was
sir.
also recalling her. She
Yeah.
that time because
Okay, okay.
that was the time those she was wanted to pursue PhD also, but she working over there.
Okay, fine. So anyway, Kiran, congratulations.
Thank you so much, sir.
And at third position, we are having Arsa Vali Ramakrishna. Are you there? Mr. Ramakrishna, are you there? No? I hope the Abhishek Nandi is there.
Ramakrishnan is also there, sir. I think he has muted his.
Ramakrishna, if you are there, just open your camera and open your mic. We want to listen you.
He might be expecting the first prize, you know. That's why he has left.
Okay, anyway, Abhishek Nandi.
Yes, sir.
Yeah.
He's there.
Tell us about yourself and about the ambience.
Sir, it's very
Since
good.
you are in MIT itself. Right.
Thank you, sir. I want to thank especially Prachi ma'am for this opportunity. Thank you so much.
Okay, fine. So our first winner, Vais Nakvi, is not present on this platform. Anyway, so this is all about the declaration of the results. I again congratulate all the participants who are who have taken part, whether they have secured any position, any rank or not, but all the participants are the winner. Because that courage which we have seen while presenting you in an online platform, so that is commendable, and I congratulate everyone. And with this, I stop here. Thank you, everyone. Thanks very
Sir,
much.
Vais Nakvi has joined. He texted me, madam, I have joined.
Okay, so Vais, if you are there, just
Guys, he is having some technical issue since beginning. So he
okay.
just texted
He is
me.
having some technical issue.
He's just about to join in 1 minute. Text me.
Till he is joining, I have one suggestion, Dr. Prachi, because the most of the paper which was presented here it was the very outstanding one, and the work literally was very good. So I was thinking that if we could have some session that they can have that at least 10 to 15 minutes for the presentation, because in the 5 minutes presentation you cannot judge anything, right, ma'am? And they cannot, that lot of effort has been taken from the participant sides also.
Yes.
So just for the next time we will have this if the such kind of presentation is there, we will keep only 5 or 10, the outstanding one 5 to 10, and then let the competition has to be between them. So the scrutiny has to be done at the faculty level, in the department level, and then if the faculty thinks that these are the participants who should go for the second round, then we can have the presentation for 5 to 10 minutes. But at least we can have some, fruitful publication as well. So this was just a suggestion.
Very right, Dr. Shalini, you are over here. Actually, being the scientific conference, I tried to give the opportunity. Yes, I have to, definitely I have to increase the number of sessions accordingly. So
Right,
that
ma'am.
the parallel session should also be there. But because of the online reason, I couldn't make it on the parallel sessions way. Because students, those who are trying to present, they want to get the presentation certificate in some international conferences and all. So far I couldn't restrict them. So far, I couldn't restrict them. And definitely your suggestion is very right that the competition should be between best one. So I hope in the next time I will be running some more parallel sessions, so the maximum participation can be taken over from their oral side, and the work will be comprehensively more clear for all of us.
And I must say, ma'am, the amount of the work which you have generated here, the platform which you have given there, because the most of the the work which the participant has shown us, it is very nice, and the lot of interaction. And ma'am, there's the like a question and answer session, and I suggest the participant has to interact with them, because
This
the
is
most
very
of the
important.
participant, what they say is they will keep silently there, whether they have joined their session or not. How we are going to know? At least they have to be present there, no? And they have to be interactive.
That is the definitely very right. You are very right, because this is for the students also, for the
Yes,
participants also. So they must have to be there, and I tried my level best to make them available, but in the online, in the inaugural
Thank you
session and the
anyways, Dr. Prachi. It was very nice, but it was learning for us as well, na? That
Madam, it is your kind words, and thank you so much for being with us, and especially JK sir spared the time, and you spared the time, and actually I'm so much thankful on my core of heart. So I think Mr. Wise Lakri has joined, ma'am. So if
Wise,
all of
yes, he's here. Wise Nakvi.
Good
Yeah.
afternoon, sir. Good afternoon, sir. Good afternoon, everyone.
Good afternoon. Good afternoon. Congratulations
Thank
for
you, sir.
securing your first position. We just want to know, because you are from this campus only, but what you are doing now, where you are, and what kind of new things or new courses you are pursuing.
Yes, sir. Actually, currently I'm in DTU, Delhi Technological University, pursuing my master's, like MTech in bioinformatics from DTU.
Okay. After MTech, you are having the plans of research, or
So as of now, so I want to take up a job after pursuing MTech, and after some time part-time PhD, no problem. But as of now, so I want to take up job after finishing my MTech.
okay, good. Congratulations again.
Thank you so much, sir.
Right. And as Dr. Salini has mentioned that the participants have get limited time of 8 to 10 minutes or 5 minutes or 7 minutes. Generally, we in a competitive presentations, oral presentation, most of the conferences which we conduct have 10 minutes time or 10 to 12 minutes time, 8 minutes presentation, 2 minutes discussion. Certainly, it is difficult for the judges to decide it, because in 10 minutes, unless you are very much close to the topic and close to the subject, it is very difficult to grasp the things. But our suggestion is also good that we can run a screening round. Like it is a 2 days conference, a 2 days symposia. So first day, in a 2 hours time, we can run a screening round, and those who qualify, at least 10-15% of the participants, they should come on the second day in a main presentation session, and certainly the time can be enhanced there. Otherwise, it is very difficult to judge the presentation for 5 minutes or 7 minutes time, right? So our suggestion is, I think we will implement and we will take it for the future conferences. Thank you, Dr. Salini.
Thank you, sir. Thank you.
So anything else, Dr. Prachi?
Sir, best Dr. Ruchi and Dr. Shilpi is going to do with their jobs, and now we are at the end of session. So I think
Thanks,
Dr.
everyone.
Ruchi,
Thank you very much.
Dr. Ruchi,
Yes, ma'am.
Please, you can continue with your concluding remarks.
Okay, sure, ma'am. It is a big responsibility you have given to me. I am audible?
Yes, we are.
Okay. So, dear esteemed speakers, those who are available right now, participants and the organizing committee members, it is an honor to present the concluding remarks for this remarkable international symposium cum workshop on futuristic bioinformatics shaping young minds, organized by the MIT Institute of Biotechnology, MIT University, Uttar Pradesh, Lucknow campus, on the occasion of the International Bioinformatics Day. Over the past 2 days, we have witnessed an engaging exchange of ideas, insightful discussions, and cutting-edge research in the field of bioinformatics. To summarize day 1, that is 20th March, the symposium commenced with an auspicious inaugural session featured addresses from distinguished dignitaries. Welcome address was proposed by Dr. J.K. Sir, Professor R.K. Sir, and Dr. Kamal Rahman Ma'am, Wing Commander Professor Dr. Anil K. Tiwari Sir, and blessings we have received from the Professor Dr. Anil Bashir Sir. We were privileged to have a Professor Dr. R. Ravi Shankar Sir, Chief Scientist CSIR CDRI Lucknow, as our chief guest, who provided valuable insight into the structural biology and bioinformatics. Next, the session started with the technical session that was delivered by Dr. Anshu Tiwari from Vanderbilt University, USA. He has discussed the microRNAs in the machine learning and also discussed the biomarker discovery, diagnosis, and also given the hands-on and shows the various tools and softwares that he has used in his research. Further, the technical session was given by Dr. Manmohan Pandey from Agilus Diagnostic Lucknow. He has given a lecture on introduction to NGS and its application, and also given a hands-on on gene expression analysis and the NGS pipeline. The day concluded with the oral presentation session showcasing a diverse research contribution from the participants. To summarize the day 2, that is 21st March, the second day began with the insightful technical discussion featuring Dr. Krishnapal Singh from University of Rostock, Germany. He had delivered a lecture on a role of system biology in target identification using disease map. Also, he has discussed on system biology approach in understanding disease pathways and drug targets, integration of multi-omics data. The second session was given by Dr. Shweta Singh Chauhan in Silico Minds, India. So she has delivered a lecture from data to drug, how AI is shaping the future of medicine. So she has given a brief overview of the artificial intelligence and how the machine learning is used in the drug discovery. And also she has shown her research work, what she has done. The next session was taken by Dr. Jiten Narayan, CSIR, IGIB, Government of India. He has given a lecture on comparative genomics of SARS coronavirus. And also he has discussed about his pipeline that he has made during his study in the IGIBI. Then a scientific session continued with the oral presentation followed by prize distribution ceremony recognizing outstanding research contribution. To summarize, the symposium successfully brought together 163 registered participants and received 45 abstract submissions. We had attendees from the prestigious universities and institutions across India, including our very own MIT University, JSS Academy of Higher Education Research, Paro University, BHU University, Tamil Nadu Agriculture University, Chandigarh University, Delhi Technical University, and many more. Participants include graduate, postgraduate students, research scholars, and faculty members, all contributing to the success of this event. So here I would like to acknowledge Srijal also for providing this registered participant data how many registered participants and different institutes were there. I extend my heartfelt gratitude to all esteemed speakers, panelists, and participation for their invaluable contribution. A special thanks to our chairperson, rapporteurs, and the entire organizing team for ensuring the smooth execution of the symposium. So thank you once again, and I look forward to future collaboration in our collective journey of the scientific discovery. Thank you so much. Now I will hand over this session to Dr. Shilpi ma'am to deliver a official vote of thanks. Thank you so much.
So thank you, Dr. Ruchi. Thank you, everyone. So good evening, everyone present here. As we come to the end of the 2 days International Symposium cum workshop on futuristic bioinformatics shaping young minds. It is my honor and privilege to extend a heartfelt vote of thanks on behalf of the organizing committee and the entire fraternity of Amity University. I would like to express my gratitude to the Almighty for providing me with the strength and wisdom of organize this 2 days International Symposium cum workshop to celebrate bioinformatics team. Without his blessings, this event would not have been possible. First and foremost, I express my sincere gratitude to our distinguished chief guest, Professor Dr. R. Ravi Shankar from CSIR CDRI, whose guidance and support have been instrumental in making the first day of the event a grand success. In continuation, I extend our special thanks to esteemed speaker, Dr. Anshul Tiwari from Department of Molecular Physiology and Biophysics, Vanderbilt University, Nashville, USA, Dr. Manmohan Pandey, Senior Bioinformatics Manager, Agilis Diagnostic, Lucknow, Dr. Krishnapal Singh, Scientist, Department of System Biology and Bioinformatics, University of Rostock, Germany, Dr. Shweta Singh Chauhan, Senior Scientist in Silico Mines, India, and Dr. Jiten Narayan, Senior Scientist, CSIR, IGIB, Government of India, who have shared their valuable insight and expertise for making this symposium an enlightening experience. Dear speakers, your knowledge and contribution have truly inspired us all. Thank you so much. I'm thankful to our expert judges, Dr. Vinay Dwivedi sir, Dr. Prekshi Garg, Professor Dr. Shalini Singh Visay ma'am, Dr. Meenakshi Srivastava ma'am, for providing the valuable suggestions to our presenters and evaluating the oral presentation. Thank you, judges. I must take this opportunity to extend a profound gratitude to our beloved founder, President, Honorable Dr. Ashok K. Chauhan sir, for his blessings, and Dr. Asim Chauhan sir, Chairperson, Amity University, Uttar Pradesh, Lucknow, for their motivation, constant support, and encouragement. We are very much thankful to our Pro Vice Chancellor, Dr. Anil Vasisht sir, Deputy Pro Vice Chancellor, Wing Commander, Dr. Anil Tiwari sir, Dean Science and Technology, Professor Dr. Kamal Rahman ma'am, Dean Academics, Professor Dr. Rajesh K. Tiwari sir, Director, Amity Institute of Biotechnology, Professor Dr. J.K. Srivastava sir, for their guidance to conduct this international event with the fruitful outcome. Further, I extend my appreciation to the technical support team and IT team of the university who worked tirelessly behind the scene to ensure the smooth execution of the program. A big thank to our past chairperson, Dr. Mala Trivedi ma'am, Dr. Aditi Singh ma'am, Dr. Gurjeet ma'am, Dr. Rachna Chaturvedi ma'am, Dr. Nikita Basant ma'am, Dr. Jyoti Prakash ma'am, Dr. Vineet Avasthi sir, Dr. Rahul Gupta sir, for their expertise, insightful guidance, and meticulous management ensured the smooth and productive flow of the session. Thank you so much. I would like to express our sincere gratitude to our reporters, Dr. Shaili Mukherjee ma'am, Dr. Sabah Hassan ma'am, Dr. Somali Sanyal ma'am, Dr. Sonia Chadda ma'am, Dr. Sujit Pratap Singh sir, Dr. Asaf Ahmed sir, Dr. Rachna Singh ma'am, Dr. Garima Avasthi ma'am, Dr. Paras Porwal sir. Your meticulous attention to detail, insightful summaries, and timely delivery of the report have been invaluable to the success of this event. Thank you so much. My sincere gratitude go out to our participants, young researchers, and faculty of other universities also, for your engagement throughout the day have been remarkable. Thank you so much. Last but not the least, I'm grateful to our organizing committee members, Dr. Prachi Srivastava ma'am, Dr. Ruchi Yadav, and Dr. Manish Dwivedi sir for their continuous support and motivation. I would also like to thank our dear student coordinators, Srijal, Shrukiti, Shivam, Tansa, Janvi, Rida, Sankalp, Farhan, and Aman for their unwavering dedication and tireless effort in ensuring the remarkable success of this event. Thank you so much. Thank you all once again and have a wonderful time ahead. Thank you so much. Prachi ma'am, please.
It is requested to all the participants, those who are on the podium, please open your cameras so that we can have a group photo.
Yes,
virtual.
ma'am. That's what I was going to say. All those who are present, they can open their videos.
ग्रेड में खोलिए ग्रेड में खुलवा दो सृजन
I think everyone has opened. Shivani Mishra. Okay, so all volunteers are there with Janvi. So they are in one frame. And with Sankalp also. Okay, so you can be in one frame. So, but all volunteers should be there. Okay, ma'am, we can take screenshots.
Thank you so much. Thank you so much to all the participants and the dignitaries for opening your cameras. Thank you so much. So Janvi, can we proceed for the national anthem?
Yes, ma'am. Yes, ma'am.
So we are at the end of session till Janvi is preparing the national anthem video. Once again, being the convener of this conference, I'm really feeling so much delighted that such eminent judges, chairs, rapporteurs, our HOIs, directors, participants, my colleagues, they supported a lot to make this a symposium, but because conduction of virtual mode is little bit tough, no doubt. And making students oriented, making participant oriented, sometimes it becomes very tough, as Shalini also discussed, Dr. Shalini has also discussed. So I would like to thanks to all the participants that till the end of the session, they didn't leave the podium and still we are active on the virtual mode. That is the big success of this symposium. And I hope the symposium might have given you certain insights that how bioinformatics are opening their roads and how it is opening their antennas, that I'm so happy that students from the different disciplines, they are putting bioinformatics effort in their results. Either they are from the microbiology, from the cancer biology, from the plant biotechnology. All of them are amalgamating the so important subject of bioinformatics. So this was the platform from where they can get the certain new ideas so that they can more exaggerate their research by putting the bioinformatics efforts. So, and definitely this is the era, digital era, scientific digital era. We all of us facing the certain challenges, certain limitations, as I discussed with most of the speakers also. And very soon we will be overcoming, and definitely we are in the virtual human physiome project. So definitely all the bioinformaticians are contributing to give you a model system, a virtual model for the scientist, for the biologist. They can perform their experiment with the sustainable environment, with the eco-friendly manner, cost-effective manner, and with the sustainable development, you can say. So very soon we will all be having, like in systems biology, we will be having our small model systems that the scientists or the researchers or the budding future scientists can utilize them. And very soon we are about to complete the human physiome project. Like everybody is knows about the human genome project. So very soon we will be completing the human physiome project, and the day is not far that when all of us were doing our science with the digital platforms. Thank you so much from my core of heart, and I'm feeling so overwhelmed that bioinformatics got the recognitions, Nobel Prizes, and acceptance, not only in core bioinformatics, but all the domains are utilizing either the immuno, cancer, plant, whatsoever. So once again, I would like to congratulate all of you and extend my thanks to all of you for being the part of this Bioinformatics Day. I hope I will be able to conduct this Bioinformatics Day in next year also with a more grand way. This is our first setup, and I'm overwhelmed that when I put the idea, it was just an idea blinked in my mind when I was reading something during the Nobel Prize. Ruchi, Shilpi, Mani, yes, ma'am, you must have to celebrate. So
Yes,
they were the
of
gearing
course.
force. And then a student like Sijal, Shivan, Janvi, Tanzait say all, ma'am, you must have to celebrate. People must know that bioinformatics is also having its own one day. So you must have to celebrate it. So I am so much thankful, and I hope in near future I can give something more valuable and more practical oriented workshops and symposium. Thank you once again from my core of heart. Thank you so much.
Thank you so much, ma'am.
Janvi, you can proceed with the national
Yes,
anthem.
Yes.
With this, we officially conclude today's event. A heartfelt
Dr.
thank you to
Meenakshi is
all
at
our
the podium, and I hope Meenakshi is expert of AI. I would love to hear from Meenakshi ma'am how AI is going to digitalize all the world.
Well,
I will love to hear from you.
you haven't left anything for me to speak. Okay.
Ma'am, you are muted, I think. You are
No,
muted.
ma'am is
Your voice-
audible. Ma'am is audible. Yes, ma'am, you are audible, ma'am.
Yeah, I was saying it is very difficult to speak after you, Dr. Prachi. But indeed, the session was excellent. And yes, there is no doubt that how bioinformatics is just changing the field of research. Okay, every day you read a paragraph and you will get n numbers of idea to proceed, especially the students who are working in self mode. But one thing, bacho, do not run behind that some researcher has used that tool or that value. So it is not required that you follow only that. Always have a comparison before. Okay, and then finalize your field. Thank you, Dr. Prachi and the organizing committee for giving us this chance. It was really mindful. Very nice sessions. Thank you.
Thank you, Meenakshi. And IT, you are the backbone of ours. So how we can forget you? Bioinformatics. So thank you so much, Dr. Shalini, Dr. Meenakshi. So Janvi, I hope we can proceed for the national anthem.
Yes, ma'am. Yes, ma'am.
Okay, thank you.
So with this, we officially conclude today's event. A heartfelt thank you to all the esteemed guests, dignitaries, speakers, chairperson, reporters, participants, and organizers for making this gathering a grand success. We hope this event has provided a valuable learning experience and inspiration for all until we meet again. Keep the spirit of knowledge and innovation alive. I request all the respected dignitaries and participants to join us for the virtual national anthem.
अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all for your presence and participation. Thank you all for your presence and participation. Have a wonderful evening. Thank you.
Now we can disperse. First of all, oh, Gayatri madam, hello, how are you? So nice to see you. How are you?
Thank you. Me and my
Doctor,
student attended yesterday also.
oh, that's great.
Yeah, yesterday also they were there, and it was very, very useful for them. For us, this field is new because we don't have a department of bioinformatics and no support. So, you know, they are just venturing into this field, and for
So
them
nice
it's
of you, ma'am.
very, very informative.
Thank you. Dr. Gayatri is the principal scientist in SIFI Bombay, ICR. And he is so much instrumental for these kind of conferences and supportive. This time she was a little bit occupied and still she attended all the sessions. Thank you so much, Gayatri ma'am, for being with us. Thank you.
Thank you, ma'am, and hearty congratulations for successfully completion of this program.
Just
Please
because
let us know your next. We'll definitely would like to join.
Definitely, I will send you all the abstract books and the required material. And thank you so much, ma'am. People like you make the conference a success, ma'am.
Thank you.
Thank you
Thank
so
you.
much.
Thank you.
So we can disperse, I think. Thank you so
Yes,
much.
yes, yes.